Bex? No, I said Ilex.
Ilex. Oh, come on.
Ooh.
Yeah, I've got a little bit
There
of delay
goes a
in the
very large
conversation
and
here, so
gorgeous
it's taking a minute
siphonophore.
to get what you've got.
What'd you say, Mike?
Oh, I know. Alan wants to play with the siphonophore.
It's gone.
I was just going to tell you a group of squid is not a squad. A squid squad is a bunch of squid researchers.
Oh.
And it is safe to call them a school of squid because they orient together and they do match all the criteria, or some species do, for that are used for fish schooling.
Fish schooling. So what's the difference between a school of fish or a school of squid versus a shoal? Shoal multiple species?
A shoal of fish or squid are a bunch that are all together, but they're not oriented towards each other.
Oh, okay. Just like a group without any real direction. Or, yeah.
Right.
Okay. Got it.
Okay, so I'm going to hang up now.
Okay, Mike. Thank you.
Cool.
Okay, thanks, Mike. I hope we need you more.
Yeah, we're going to find more squid, I promise.
Ring him up on the squid phone. Did you ever read that New York Times article about him getting called up on the squid phone?
No.
That was like in the title of the...
Really?
Yeah. It was something that Chris Moss said, and he was in following something and they called Mike to see it.
And it was in the New York Times?
Yeah, the whole story was written up in the New York Times.
I'll have to find that one.
Yeah. It was a behavioral observation from the ram's horn squid, which was seen in situ, and so the video was really valuable because it explained something about how this squid behaves. Actually, I think they got a paper published in about 30 days.
Oh, that's impossible.
It was so fast. I don't know the exact number, but it really was.
Shenanigans on that. That's ridiculous.
I'll look it up for you later. So we could just know for sure when the sighting was and when that paper was published. Now we're getting there. See in the bottom now, or is that fake?
No, that's real. And it's sandy. So you have some bioturbated sand.
See those little burrows in the sand?
Mm-hmm.
Those are large.
So I think we're going to be getting an eDNA sample at the bottom when the ROV pilots are ready. Usually we take a few minutes to settle in before we start moving. So we'll get to the bottom, let the ROV pilots line themselves up, and then we will take off.
Yep, looking forward to it.
There's something there at 9. Spiky thing. Probably detritus in these little pits here. But these pits could be formed from what, underwater fauna? I mean, under sediment fauna, like worms or in fauna. They're in fauna. I have to do a box score to find out what's in there.
Are we set up for that?
No, we do not have a box scoring capabilities. Can we do sediment cores? I don't think there's sediment cores on our. We could do. Oh yeah, I guess technically could.
Mm-hmm.
We could sample it with a suction hose, but that's not. That's a little different than a core, right? Where the core would show layering down. There you go. Sometimes there's little snapping shrimp or Acanthocaris living in these, a blind lobster. We also will have tilefish or sand tilefish, golden tilefish. They like to make the big burrows in the sediment. But I don't see anything that big yet.
No, so
No.
Mike Vecchione confirms that paper that
30 days.
squid was observed 27 October and the paper was published 27 November.
You guys are monsters, man.
That was in the journal Diversity, by the way.
Paper writing superheroes.
Yep, paper writing superheroes.
$16.22. Thank you. So that was eDNA sample number 2.
Oh, we have our second sample of the day.
Yep.
See, the cameras are tilted down so we don't see too many of the jellies.
Your midwater stuff.
So we'll be getting set up, and then we will be traversing up the Scarp.
Ooh. Oh, neat shrimp. It's kind of on his side.
It is. It's
Oh,
tilted away from us.
Fish behind him. Oh, hatchet fish.
Hatchet fish.
Cool.
Indeed.
He's the first one this cruise. Very shiny.
Easy to see how it gets its common name, hatchet fish.
that? What is that?
Moral?
Nothing.
Oh no!
What do you think
I
that is?
think that might be an isopod, right?
Oh,
A
yeah,
swimming, one of the
look.
swimming isopods.
Yes, I agree with you, an isopod.
Nice. Yeah. I've seen occasional videos of these.
They
And
must like jump off the bottom or something.
Yeah, they
Or
can
usually-
swim with their very long legs. It's actually quite beautiful. It'd be nice if this guy gets up in the water and we get to see him. Oh, landing.
Disappears immediately. It just
Oh,
looks
it
like
is. How many of them?
Yeah,
Thanks.
we're not seeing.
Thanks for following that. Yeah, how many others are out there? That was really cool. I guess they're commonly referred to as water walkers.
Quite a bit of bioturbation here.
Yeah.
The little mounds and we're out there in that last field of view. There's a little particles of like I don't know, something eating the bottom. You could see their feces or pseudo
Yeah.
feces.
Yeah, I saw that.
Yeah, that looked like a sea cucumber poop.
Mm-hmm. Got something swimming at 3 o'clock. Maybe a shrimp. Any idea how far away from the wall we are? Not too far. Oh, something silvery.
Fish of some sort. I think.
Yeah, looks like it could be.
Is it probably an eye reflecting back at us, you think?
Could be or a photo for. Maybe a crinoid at 9. Yeah, a little crinoid on the bottom.
Ah, it's interesting.
Yeah, I don't usually
Didn't
see
know
them
how
on
often
the
they're
sand.
on sand.
Yeah.
It looks sadder on sand than when it's perched up on
Like you got
the
lost
edge
or
of a
something.
coral.
Yeah.
Something in the water column right ahead. Small. Yeah.
Little shrimp.
Oh, yeah. Suggested, I guess.
There's a little bent antenna.
So pretty, delicate.
Do you know how far away we are from the waypoint? The bottom of the wall? Roughly. Go ahead, Nev. No, the base of the wall, like until we start seeing some relief.
Okay.
Okay. Yeah. Okay, thanks.
Guess that's a nice little larvacean house. I don't see moving in there. So it seems like there's something in the middle. I was just sort of staring at it, trying to figure that out.
What is that?
Ooh, yeah, let's look at that.
Same thing,
Oh.
huh? Larvation house?
Yeah,
maybe?
an old one. Yeah, I think so. Did you see Scott France's note in the chat about how we might encounter a black coral that branches from the base, and so it might appear to look like a crinoid.
Oh, okay.
Yeah. We definitely saw a crinoid.
Yeah. Here's some nice bioturbated holes in front of us. They look pretty active.
Yeah, I agree that last thing was a crinoid, and I wouldn't expect to see the black coral until we actually got to the wall and some hard substrate. But
Yeah.
I thought it was interesting looking at that crinoid, how much it reminded me of this previous black coral that we'd seen at these depths, and we collected back in 2017.
Squid.
Oh, there's the squid.
Thanks, Scott.
Now I'm guessing that's
Ilex.
Ilex, but we'll get some confirmation soon. Wow.
Awesome.
Looks like it.
Mm-hmm.
Thank you. Oh,
There's something in the sand there. Looks like a fish with his tail up. What are you doing, bud? Little rat tail?
now is it? I wonder if it's.
Looks like he's stuck.
I wonder if something's got it. No, a hatchet fish coming straight at us.
Excuse me, we should be looking at me.
Hello, look at my eyes.
Sure.
Yeah, I think so. Yeah.
Oh yeah, I see him at 9 o'clock.
Yeah, it's Mollusk Monday.
Mm-hmm.
Oh, now that's interesting.
Oh, neat. You can see his suckers. Oh, he's changing color. Ooh.
Doesn't have to go too far away. I love that stance. You
Like
know,
up on his
resting
elbows?
down, yeah, on its arms.
Is our fish still there? I hope he escaped. There he is. Uh-oh.
George Matsumoto in the chat is one potential explanation is that it was sort of blinded and sort of took a nosedive.
Mm-hmm.
But it's still there when we didn't have the lights on it. It's interesting.
Hi.
Hello, Okeanos.
Hi, who's this?
This is Mike.
Oh, hi, Mike. I don't know why you sounded different.
Oh, there it is.
Hi. I was just trying to type into the chat that the hatchetfish are sternoptics. That's the
Sternoptics.
genus.
Thank you.
When that
Thank you.
that ilex showed up, and just wanted to let you know that it's really common for them to go down and sit on the bottom in slope depths during the day.
Are they just hanging out there, or is it just a behavior?
Yeah, basically, there are several species within that family that are associated with the continental slope and outer shelf, and all of them do that.
It's like hanging out on their elbows.
Mm-hmm.
They need iPhones. They've got to do all day long.
Look, The reason they're up on their elbows is because the funnel is on the bottom. It's on the ventral part. Mm-hmm. So they still have to breathe. So up on their elbows, that funnel is up out of the mud.
They're not
And they
suffocating
can,
themselves.
Makes
inhale
perfect
around the
sense.
head and then exhale through the funnel.
Awesome.
Really cool.
Keep their face out of the dirt.
And as you saw, that one was flashing its chromatophores pretty actively, and you could tell that it's alive. That
Yeah,
way
it was neat.
before it took off and swam away.
Ooh, we got a red crab.
Yeah.
Chasion, maybe? Yep.
Oh.
It's Quincus bifera. The red. Quincudens. Yeah. Chasion quincudens. Because we also have the golden crab, which is Chasion fenneri, which is more on the east coast. There is a fishery for those. Ooh, squid, yay!
No, look, it's like a patrol.
Now you're starting to see a squad of squid.
I think I see Bathynomus.
See, they're
or
all
fish.
swimming in the same direction, lined up together. That's one of the characteristics of a school.
Yeah. Well, they all like kind of the same purpose. Is that Bathynomus? Yay, Bathynomus! The giant isopod.
Wow.
Can you put lasers on him for me? Oh, he's like 15, 20 centimeters. Bathynomus, probably gigantea.
Well, I'm going to have to step away for about 10 or 15 minutes, so it's time for the squids to show up in numbers.
Of course, definitely. Thanks. Awesome.
Bye.
Bye, Mike.
Is that a collection?
No, we
I
are
don't
not
think
collecting
so.
that. Get out of here. We'll be here all day chasing him down. So this is Bathynomus giganteus. It's a giant isopod. So they're sort of related to that we just call them pill bugs.
Pill bugs,
Or
yeah.
roly-polies. They're similar. But these are huge.
You can find them in your garden.
Pretty cool.
Definitely has a swimming morphology on that tail.
Yeah, and they'll actually pop up off the bottom and use their swimmerettes underneath to escape. But they're mostly bottom dwellers. A little hatchetfish or a little fishy in the front.
We were diving with the GSL sub in a sinkhole up off the Keys, off around Marathon. And the top edge of the sinkhole was this consolidated clay, and it was just riddled with holes. It was like a condominium for these Bathynomus.
Oh, nice.
And each hole had a Bathynomus in, and occasionally they would swim out over the sinkhole in formation and then go back into the hole.
Oh, that's
Really
cool.
crazy.
That's
I don't think I've
cool.
ever seen them swim. I would love to see
There's footage. We have some highlight. One of the highlight
Uh-huh.
reels from my last cruise had
that.
them.
Nice.
So it's right on the NOAA website. And that would be winter of 2019. Yeah, they do look upset, don't they? The first time I saw one of these in the jar, Chuck Messing picked it up out of the jar and put it on the side, and he was like, "Luke, I am your father." It looks a lot like a Star Wars character. Pretty neat.
So nice. They have plush toys of them.
Yes, they do, and I want one desperately. Like right now. I know what I'm shopping for after the cruise. Oh, another squid.
There goes another squid.
Oh, there he goes. He's swimming.
There he
Get him,
is.
get him.
I got to see it.
There he goes. He's swimming.
That's good.
Yeah, don't break your tether. My gosh. Maybe a
Too good.
tripod or something? What's this fish here at 9 o'clock? Looks like a tripod fish. He's not very far off of the bottom.
Ooh, I
Yep.
want.
Yeah. Looks like his tripods are buried, though. Bottom's too soft.
You can see how it's sitting and waiting for food to come along. Has all that.
Like modified pectoral fins, I would
Sensory.
imagine, right? That's what these are.
That was nice. There goes, I think, another cyclothone, which is what we saw over top of the isopod.
Lots more of these bioturbated holes. Looks like maybe sargassum, dead sargassum detritus in the hole here. More ilex squid.
6 right there. There's 7.
I don't know. They definitely look like a squad.
Ooh. I think they're a squad too. We were having this, we were having this debate.
Curious what tripod fish that was.
We've seen that one before. Yeah.
You think it's that? The four-finger quadrophilus?
Maybe. Bathynomus?
I don't know.
Terios?
Yeah, Andrea Quattrini says it was probably Bathynomus,
Bathynomus.
CF quadrophilus.
So similar to.
Yeah,
That's what CF means.
if there was no subcaudal notch.
Look, we got
What do we got?
That's looks like a little white hexagonellid and an ophiuroid. But the ophiuroid was like sand colored. I highly doubt that was
Lots
a
of
hex.
squid.
I
So
don't know.
many of this ilex.
You can see the increase in the slope. That's promising for getting to the bottom of the wall.
Yeah, we have confirmation on the identification of that tripod fish. Bathynomus quadrophilus.
You might have to change the name from Tortugas Valley to Calamar.
Indeed. A couple things in the water column there. Maybe another larvacean house. Yeah, that's good enough, I think. Just a little larvacean house. Also maybe Dunton. I don't see the animal inside it. What's that dead ahead on the bottom? It's got a fish, yeah. Yep, we can keep moving.
It seems like this region has a lot of rich organics in the sediment. So I guess that's a lot of good cause for these animals to dig in there and turbate it up. And the isopod cleaning out the organics from the actual rock sediment and squids and such.
Another one of these ilex squid cruising across the bottom. This
Flying
time
along.
heading towards us.
Great shot. Oh my gosh.
Wow.
All the chromophores on its side.
Continuing to make our way up slope to what we think will be more steep, harder bottoms, but we don't know. Someone join us? Oh, no. I keep getting warned in the chat that Scott France is going to leave, and the next beep I'll hear is going to be him leaving. and then it happens anyway. So evidently this is Mollusk Monday. So our little contribution to Mollusk Monday is a whole bunch of ilex squid.
Is that a Twitter thing?
Yes.
Oh, okay.
Yes. An invertebrate every day of the week. Wow.
Fascinating.
Yeah, it's really just resting.
Such an interesting leg arrangement.
Mm-hmm.
Tentacles.
Hello, Okeanos.
Hello. Hi, Mike.
Hey, I was just going to tell you that there's two species in that area that are that this could be: Ilex coendedii and Ilex oxygonius. Wait a minute, let me turn off my speaker here. And the only way to tell between the two of them is to catch a mature male and examine closely the details of its reproductive arm, what's called the hectocotylus, which is modified for transfer of spermatophores to the females. So I can't be confident as to which of these species we're seeing.
Okay.
The one that we've got a close-up on right now doesn't look like it's quite mature enough to spawn. Usually you can see development of the reproductive organs through the mantle musculature at the posterior end, back between the fins. And it doesn't look like that this one is completely mature yet. But this is the right place and generally the right season for ilex to be spawning in this area.
Hmm. Wow.
The white mass at the pointy end there, that's reproductive organs. But it doesn't look quite mature to me yet. The way that the shape of the head looks like it's a male. But like I said, you can't tell very much without examining the details of the hectocotylus.
Unless we catch one.
Yeah, catch about 50 of them, and then we'll figure out what they are.
Scott? Hey, Scott, do you know the field of view of these ilex? When they're swimming, forward, can they see 180 degrees forward, or are they just seeing to the side?
My guess is that they can see pretty much, they're actually swimming backwards. So the pointy end is the posterior end, and they can see pretty much 180 degrees on each side with the two eyes.
All right. There we've got a nice...
They can orient easily towards something that's forward of them.
Nice. Asteroid, starfish, very long, thin arms.
Okay, I'm going to hang up now until I think of something else to say.
Okay, Mike, thank you very much.
Bye.
That's nice. I think we have Chris Ma in the chat, so he might be able to give us a feel for what this star is. Well, yep. Does look similar to the one we saw before. It's funny to see it. Looks sort of sunken in the sand in the front there a little bit. Must be the sediment must be very fine.
Oh, it's a really great view of the specialized fin rays.
Hey, Alan.
Hey, Chris. Dr. Ma, how are you?
Good. It's always a good Monday that starts with the Bathynomus.
Indeed. Tell us about that star.
Well, I think there's one of two choices. It's either Henricia or something called Zoroaster. It seems odd that it would be Henricia just because we're so far away from any sponges that it might be eating. So that's why I think, especially given the sediment bottom, it's a very commonly encountered deep-sea star called Zoroaster fulgens. These are kind of, I don't know how to put it, sort of very old-looking sea stars that belong to a strange family called the Zoroasteridae. And they have little spines that are located around the underside, and they dig little holes into the sediment looking for like clams and snails and things. And they didn't actually, they turn up in collections all the time, but nobody ever has actually done much with their biology. So, like looking at their gut contents, looking at their looking at them alive, people thought they were suspension feeders for a while. So this is one of the valuable things that these ecological observations give us, is some context of what it is they do, even though we know they exist.
Thanks, Chris. I also want to just let you know, if you can see, there is a sponge present out on this sandy bottom. This is a glass sponge. Looks to be growing on top of either a dead stalk or maybe a stalk it's made.
Yeah, it's interesting. It looks like there's some sort of polyp on the bottom with a Galathiid clinging on, and a brittle star as well.
It was definitely things that look desperate for hard bottom, right? There's covered with polyps, the Galathiid, or the Galathioid anyway, and then brittle stars in there as well.
Those are almost certainly Ophiocanthids. Also, just thought I would mention that we're seeing a whole bunch of these little fecal mounds, these little poop piles, likely left by sea cucumbers or other kinds of worms like acorn worms. Those are always, that sounds funny, but it's always cool to get observations of those just because they're so orderly. And those are actually a valuable biological trace, because a lot of the worms and the sea cucumbers that process them, they aerate the soil. They contribute to bioturbation in that way. So seeing those is always worth a shot, just because, they have that interesting story, and just because, well, I don't know, I think piles of sea cucumber poop are kind of cool. That's just me. Anyway, I will go back and look at the picture of the sea star and sort of mull over it further, but I'm sure we'll see more as we progress.
Sure. And that stalk sponge...
Thanks again, man.
Nolan Barrett points out, and it seems reasonable hypothesis, that that's a member of the family Hyalonematidae. It's just one of the families of glass sponges that have developed a stalk to get them off the bottom, where the...
Get away from that ever-present sediment,
or
trying to
into the
consume
current.
them.
Yes, exactly.
Yeah. Looked like we had our first sighting of some hard substrate up ahead. That rock protrusion.
Yeah, at least semi-hard, more consolidated.
Yeah.
I'm thinking back to what John Reed said at the beginning, and that they had, poked some of this, and you could, see it fall down. It does look soft like that, but...
Well, there's another squid.
Mm-hmm.
I like hanging out on the bottom.
Is that squid ink in the water, maybe?
Like the sea snot?
Yeah.
I don't know what the final product of squid ink, after its initial release, what happens to it. Our Global Foundation for Ocean Exploration's pilot team is going to do a swap, so please stand by as they brief out the next person, and they hop into the chair and take us along our way. It is lunch on the Okeanos Explorer right now, so everybody's trying to jump in and out and get a quick meal in.
As one does. Getting our first look at hard bottom, or harder bottom than the sand.
Yeah, there's still a little protrusion there, but you can see the rise that created it behind it.
Mm-hmm.
Looks like it's been sedimented over. Definitely a porous material. There's a lot of little holes in it, and most likely like a mudstone.
I think maybe keep moving is not a bad idea. Just to, yeah. Ooh, there's something there on the edge, though. Maybe we could have a quick snap zoom of that. That you're pointing right, yeah, directly.
Right at the lasers, yeah.
Yeah. Yep, thanks. Oh, yeah, glass sponges for sure. We got a bunch of Pharea. Ooh, yeah. Oh, and someone's sticking out of the hole. I wonder if that's one of those blind lobsters.
Wow. All those little pores holding a lot of life right now.
No, we don't need the lasers right now, thanks. Wow. This is on our collection target list. If there's a quick sample to be made, if it's convenient. Not sure how that lines up for you. Oh, I apologize. Let's skip that sample request. Let's look at what's to the right here on the wall right in front of us. Yeah. Thank you. Yeah. Oh, I love this animal.
What is that? Looks like a millipede.
It does look like a millipede. I would suggest that if we're going to collect here, that this is the animal to try to suck up. This is great footage. Yeah. So
this is Watchley. Can we get a collection of this?
Ooh, yeah, I guess it's a scale worm, but
Thanks.
It's really quite beautiful.
Looks really cool. All right, we're going to try for a collection of this beautiful scale worm.
Pollinoid scale worm, yep. This is a polychaete group that has scales over at the top over the back of it. In this one, they're very flexible.
Is it the same species we've been seeing, you think? Okay.
All right, so our pilots are going to use our suction sampler to try to slurp up this scale worm. It's pretty exciting.
And it's going to be sample number 3B.
3B.
B for biota.
I already have 2 water samples for our environmental DNA analysis, and now we're going to get a biosample.
So if this is a scale worm, it might just take off.
It might, yeah. We'll see how good that vacuum cleaner is. This reminds me of our old Electrolux back in the day.
Right. Did it pick up Aussie hair? That's what I need, one that picks up Aussie hair. And I'll tell you, that they shed. Industrial strength. All right, so it's like 6 o'clock of screen right now. Looks like a squishy purple.
Yep. That's it. I feel like we're on the other side of the camera in a horror movie, sneaking up on it.
Oh, there's the pycnogonids on that rock, too. There's 2 of them.
Oh, good eye, yeah. We looked at the upper part, but those were hiding underneath.
Mm-hmm.
And we thought we saw one of those blind lobsters hiding in a little hole.
Acanthocaris?
Yeah.
Here's a... What is this? Did you look at that? Shrimp? Pink shrimp?
I think that's what
As
we
thought
thought
it was?
was
Okay.
one of the blind lobsters.
Acanthocaris should be stark white, so I don't know, maybe a
Ah.
different species.
Okay.
Or genus.
Oh, another squid flying
Yep.
overhead. Staying put so far.
Yeah. And some of these worms are really toxic. Like fireworms and stuff, you can touch them and it just burns your skin.
Oh, wow.
So we'll have to be really careful when we manage this
Break
thing.
out the big gloves for this one.
Yep. Oh, yeah, you can see his scales. And there he goes.
Wow. That's a nice demonstration of how crumbly and weak this supposed rock is as well.
That's what John was saying. It's real soft.
Oh,
It's
wow.
really, it's like not quite solidified into sedimentary rock.
I wish everyone could see the actual collection chamber. We could see it swimming
see it
around.
on stream 3.
Oh, it's on stream
in the quad
3.
split.
You can see the scale worm's legs beating and swimming. It's beautiful motion. Oh, shoot. Oh, crap. Oh, man.
It's 1710 is the... What do you need help with? Did you get it? Okay. And before you speed off, can we look at these spiders? See spiders at 9 o'clock on that rock? There's a bunch of those little red guys in these rocks up here. Shrimps or lobsters, whatever they are.
All right, I'm going to go get lunch.
Okay. there's your, yeah. Picnogonids, there we go. So this is a sea spider. We've seen a few of these this cruise. I usually don't get to see them very often. Not quite focused. There you go. That's awesome. Okay, whenever you're ready, I'm happy. Thank you. Mm. There was another one just above it, and then slightly to 9 o'clock. Oh, and you don't have to if you don't want to. It's up to me. Go for it. Do it. Yeah. So there's actually two here. So it is at 8 o'clock. So down, and there you go. That's his legs right there. He blends right in, doesn't he, with all that? Yeah, it looks like the stuff. So the males carry the offspring, and we got footage of one last time I was on the ship in 1907, the cruise 1907, and it was carrying babies. Awesome. Thanks, guys. There's also like maybe a Galatheid, and I'm not sure what this red meshy stuff is. Maybe a dead hexactinellid. Has that same kind of texture. It almost looks like fabric. We also have a few Demos sponges, and then this stuff waving around at 6 o'clock is bacterial matting. And then some blue sponges, too. Some hydroids up on the upper right, and at noon. Another crab. And a crinoid. Wow, there's a lot of stuff there. And yeah, go for it. I'd like to start making our way up the wall as quickly as possible. I know that's, within reason, but yes, stop and look at stuff on the way, but so these rocks are similar to the ones we were seeing in that sinkhole, really almost like pockmarked. It looks like another Bathynomus at 2 on top of that rock over there, off in the distance. Sure, you can zoom on that. Snap zoom him. About the same size as the last one, about 20 centimeters long. Yep, Bathynomus. A giant isopod. Awesome, thank you. There's a shrimp, too, right in front of him. I think I see some squid floating above the bottom. This looks pretty sedimented, with just a few of these like cream-colored outcrops. Again, back on the bioturbation, you can see the mounds and the dips where in fauna is living. There's some more squid. A little bit of a slope. So we're going to put in a little bit of a ship move. So if you're just joining us, we are actually off the dry Tortugas today. We had to escape a little bit of nasty weather on the Blake Plateau. I think one of the predictions was 25-foot seas, and I don't think any of us want to live through that. So we came down south on the south side of the Keys. We're about 30 miles from the dry Tortugas, and I think 70 miles southwest of Key West. And we are diving this escarpment, or soon to be escarpment. We started at the base of the wall, and we're going to moving our way towards the base of the escarpment. And we've only really seen mostly sediment this area with a few places of rock, but there's been quite a bit of fauna with Bathynomus, or the giant isopod, some ilex squids. We collected 2 water samples and 1 scale worm so far, so 3 samples so far. And let's see, at the base here we're about right now we're about 975. At the base of the wall is about 1,000 meters. The top of the wall is about 820 meters. And that's a total rise of 180 meters from the base to the top. So we're trying to move along our transect from the base of the wall up to the top of the wall, and then possibly along the edge of the wall. And that way we can see multiple changes in habitat, hopefully. The highest slope on this wall is about 45 degrees based on the multibeam, but the resolution in the multibeam may not pick up local highs and local slopes, so it might get steeper in some parts. But we are still coming up on the sediment and making our way to the base of this wall. Hi, Alan.
How's it going?
Good. How was lunch?
It was delicious.
Yeah.
Yeah.
The food on this boat is awesome.
How many worms did I miss while I was
None.
gone?
None. There was no other worms. You did miss some squid.
Oh, nice.
And 2 pycnogonids. Ooh. Can we zoom in on that? Looks like a sea pen. Yup. There's a little sea pen. So sea pens are in the phylum Nadaria. They're related to corals. You can see they have similar polyps to a lot of the gorgonians we see. Sometimes these, if you get too close, they pop into the bottom, so they'll suck themselves into the tube. Some species do that, not all species, and honestly, I don't know which ones do and which ones don't. That is a question for probably Scott France. But if we were to poke it, it might pull itself in.
Zip on in there.
Yep. I see another ilex squid at noon. Yep.
It does indeed.
Oh, it is a fish, not a squid. Little rat tail. Pretty small. It's less than like maybe 10 centimeters long. He looks different than the one we saw before. And he's not an Itsumia, where we were seeing on most of the rat tails were in Zumia all week.
Right.
And rat tail's probably not even the common name for them. He's about 15 or 20 centimeters. Coming up on this local rise. Looks like it's going to drop off again, maybe. It's pretty barren here. What we aren't seeing is the in fauna, which is creating all these bioturbated mounds and holes. So there's probably quite a bit of life living inside the sediment, but not much on the outside. Ooh, more Elix squid. They're swimming into school. There's a lot down here.
I love how they're often parallel to each other and sort of spread out in a line.
Yeah. You got more of these like raggedy rocks, right?
Really soft looking.
Like not quite sedimented into solid rock yet. They're really, the one we picked up in the sinkhole that looks a lot like this was almost muddy or like a clay. So my guess is these are going to probably be similar. If we were to touch it, they'd probably just break apart.
What?
Yeah, there's something there in a little hole at 6 o'clock. Looks like a little white ring. If you get a zoom on that. Right there.
Yep, dead center.
Oh, one of those little lobsters or crabs.
Yes.
He's probably a lobster. These are pretty common out here in the rock. Usually we see Acanthocaris makes their own hole, but maybe he dug this since this rock is not that hard to dig into.
I guess these are rock lobsters.
Rock lobster. I'm going to need another shot of caffeine if I'm going to keep up with you today. Ooh.
We don't want me singing. My kids, yeah.
I know, I'm a terrible singer too.
Yeah, let's go. So we have made it all the way to 977 meters.
There we go. What is going on there? It looks like a Chrysogorgid. And then
Tucked in there, and then a large
fan sponge
with a hermit climbed up on it.
and a crinoid. And an isopod, amphipod, and a crinoid.
And an amphipod.
Yeah, it's a fan sponge of some kind. Ooh, and Faria.
I think we should keep moving.
Yeah. Oh, we have a Bathypathes maybe at 6. That's a black
Yeah, you want to
coral.
take a quick look at the Bathypathes?
Yeah, if you guys want to snap zoom, just a quick ID on him. Probably what it is. Yep, Bathypathes. Thank you. This is a type of black coral. That crazy rock sticking up. So yeah, you can see the slope here is what, 30 degrees
Definitely
maybe.
picking up.
Still sedimented. Bioturbated sediment. Got something in the water column flopping around. Oh, is that one of those butterflies? The sea angel?
Maybe, I'm not sure.
We have caused pig pen sedimentation. Excuse our mess.
Fortunately, we still have a serious view.
Mm-hmm.
Oh, another squid. There's the ilex taken off.
It's constant companion on this dive, the ilex dive.
There's some more at noon. Just there we go. Just over the hill. Mm-hmm. Yeah. Mounded sediment.
We can get across this and make our way up the next slope.
The squid like it out here on those beach, on the beach. There's lots of these mounds. I wonder who's making those. They're all similar shape and size. It's probably the same critter species or something similar. Off the Keys, sometimes we find snapping shrimp that make these little holes, but I think we might be too deep for that.
Nice.
Hello, Mr. Squid. Oop. So it looks like those might even be connected, these two holes here. Sometimes they do that where they have a front door and a back door, depending on what's digging the hole. We got maybe another scale worm at 3. Hey,
on the ground.
Can you swing 3 o'clock? Yep. And there's like a longer, right, that guy, right there. Yep. Another
That's
scale worm, maybe?
pretty big.
Yeah, does that look like the one we collected?
It does look a lot like the one we collected. So this is another view of it. Wow. No, I'm not sure it is the same though, right? Didn't we see the scales more obviously separated one from the next on the other? Wow. Can we get the front end, the head end, which is down to the left, a little bit closer? See if we can look into its eyes, or where its eyes would be if they have them.
Eye spots.
All right, well, I like that.
So it's like really
seems to be covered in the sort of a gelatinous.
Yeah.
I assume it's another pollinoid, though. Scale worm. Thank you.
So 10 or 15 centimeters long. That's a pretty big worm. Yeah, we're good. Keep going.
Yeah, we want to keep moseying along.
Yep.
Can you tell from the sonar how far you are from the more vertical wall?
I'll check. Hey, pilots, do you know how far away we are from the possible wall? Yeah. Okay. Okay.
Stream 3 has high back full screen, what he's describing.
John, were you able to hear all that? If you're not listening to Stream 2 or Stream 3, I don't know if you can hear the pilots or not. But they were saying there's nothing on sonar so far that's showing it. But if you look at HiPAC, you can see how far away we should be from the base of the wall. And that's on Stream 3. But also, go ahead.
On that view of 3, there's a waypoint 2.
Mm-hmm.
Is that waypoint 2 at the wall?
Do you know where they put waypoint 2, Alan? At the base of the wall or the top?
I think that was at the top.
It was at the top. Waypoint 2 is at the top.
Yeah. I can check at the depth real quick.
835 meters is waypoint 2. So it's at the top of the wall, John.
It looks like about 900 meters. It starts the 10-meter contour lines start getting closer together.
Mm-hmm.
So that's probably 900 near the base.
Yeah, we're at 973 right now. So we got a little further to go. And the co-pilot was saying it looked like on their data that should be around 930 meters, and we're still at 973. So it's a little off.
Okay.
Another little fish here at noon. Oh, that looks funny, huh?
Let's take a look.
Little hatchet fish. No, what is that? He's cool.
I've seen that.
Little silvery guy.
Thanks for getting that.
Cutthroat eel at 9.
So this is softer bottom, more soft bottom than you expected?
I don't know, not necessarily at the base.
Mm-hmm.
I kind of expect the sediment to be at the base. I'm surprised it's so hilly. That's kind of odd. It's almost like underwater sand dunes. Which, of course, I mean, unless they're humongous, it's not going to get picked up on the multi-beam.
Right.
Just because the resolution isn't fine enough from the distance they're using their sonar at.
Ken Sulak in the chat suggested that fish that we took a good look at. It's probably the genus Polymyxia.
Awesome. Yeah, so I think we're putting another ship move in to go. Is that what I heard?
Yep.
To go closer. The bioturbations is everywhere. We got something silvery in the distance. Yep, right over the edge of that hill. It's dead center now. Right there. Yeah, it is really far. Yep. A little fishy.
Mm-hmm.
Might even be the same individual we just saw. Oh, I see another fish. Really white fish. Oh, it's its shadow. It's the same, yeah. At 4? Off screen almost?
Oh, yeah. It's a fish.
Oh, okay. Okay, no worries.
Thank you.
Yep. That happens.
Yeah, a little.
The ballet that is
Exactly.
the Okeanos Explorer diving.
Can't get every bend and twist just right.
Bad yoga move. All right, so we're having a little bit of a technical problem. Just some patience is all. We can do... Oh, Elix. We got another squid here at 3. Your deep sea desktop background. Most of the bottom of the ocean actually looks like this, just sand, as far as the eye can see. So we're just going to pause here for a few minutes until we get our technical problems worked out. So if you're just joining us, I'll do a recap. We are 30 miles off the south side of Dry Tortugas, which is off the Keys, and about 70 miles, I think they said.
Yep, 70 miles from Key West.
From Key West. We're diving. Our deepest depth today was about 1,000 meters. I'm not sure if we made it all the way down to 1,000 meters.
It was about 980 meters.
Okay, and right now we're at 973 meters. And when we got to the bottom, we saw this basic landscape, bioturbated soft sediment, and then we're coming up along these like what amounts to be underwater dunes, making our way to the base of a wall. The base of that wall, yeah. And we shouldn't be too far from it now, but the top of it is about 820 meters in depth. And the total rise we were shooting for today is about 180 meter rise from where we are or where we landed this morning until we get to the top of the plateau. The highest slope in this area we think is about 45 degrees based on the multi-beam. And the length of the feature is 920 meters.
Yeah, although...
Yeah.
Unclear. A lot of that was at the top. So.
Okay. We have done 2 eDNA samples, one when we hit bottom and one at 500 meters on our way down, and then we have collected a scale worm, which is going to be our 3rd sample for the day. So right now we're just on top of this sand dune, which is bioturbated, and we've seen a lot of squid. So they're really common down here. A couple of species of fish. I saw 2 pycnogonids, a few hexactinellid sponges. We saw some rock, some hard bottom.
Some hyalinemids stalked.
Oh yeah,
I
missed that one.
Yeah.
I saw the Faria. I used to saw that while I was at lunch.
We saw some Faria, yeah.
Mm-hmm.
Faria.
A couple Bathynomus, which is the giant isopod. What else do we see? Lots of squid. I think squid's the most dominant species down here.
For sure, the most dominant was this ilex squid.
At least from what I can see. I mean, there's probably a lot of stuff inside buried in the sand, the fauna, but we can't actually see the little critters. So
these lobsters that are made holes inside the soft rock outcrops.
Yeah, so we're just waiting now for our pilots to fix whatever they were fixing. Good. Okay. So once they get fixed, we'll keep moving forward.
See if we can make our way up the side of this feature.
Mm-hmm. Oh, yep, and we're in the Tortugas Valley, which is actually south of the Dry Tortugas. And to the east of us is the Agassiz Valley. So we had come off the Blake Plateau because of weather and sea states. And if we come off the south, we came off the south of the Keys to basically hide from it. Knock out a couple dives down here while we can.
Came right down the Florida Straits to the western end of the Straits, close to the Gulf of Mexico.
Mm-hmm. And you see another ilex in dead center. You can see him up on his elbows, I think. He's pretty far away.
So we have a request to look a little more closely at some of the cutthroat eels when they are there.
Oh, maybe not. What is that? Yeah, that's a squid. I thought for a second maybe it was the armored sea robin.
So we're just ironing out some technical issues before we can proceed farther across this sand and get to hopefully a bit of a steeper part of this transect. Curious what that is in the water column.
I'm going to guess squid ink.
Ah, thank you, Roland.
Just a video guess, though. I've been watching it.
See one of those cutthroat eels stationary on the bottom. Hopefully we get this sorted out. Maybe we can take a little closer look at that. More squids on patrol went by. If you're just joining us, we're having a little bit of technical issues moving the ROV around, but we're working those out.
We got some more squid coming up from the left. And it looks like a ctenophore, too, in the water column. We are still waiting on the ROV pilots to give us the go-ahead. They're troubleshooting some stuff right now. Yeah, go ahead. Okay. Before you speed off, at 3 o'clock, there is a cutthroat eel. Yeah, below the jelly. Yeah, you can zoom on that jelly too if you want, but no, the cutthroat eel. He's going to be at 6 o'clock on the sand. Yeah, sure. Yeah, it's a ctenophore. You can see his comb rose. And of course, we see an awesome ctenophore as soon as Alan goes to get coffee. Oh, neat. Look how beautiful he's reflecting green almost. Let's see. See if that cutthroat eel is still around. If not, we can just... Oh, there he is. Okay. He's coming into center screen from the left. Right there. Yep. I don't know. We'll see. Both. Mm-hmm. So we had a special request from landside to take some zooms on the cutthroat eels. All right, Ken. Ken Sulak, if you can hear. If you're still there, here's your cutthroat eel. Awesome. Thank you. Thank you. Some bioturbated sediment again. We've got a little mound and a little divot. And there's some larger mounds coming into view from the right side. Yeah, go for it. Okay. Hey, someone call in?
Hi, Stephanie. Scott Friends.
Hey, Scott.
Back from my lecture and joining in case we see anything cool.
Yeah, we've already seen cool stuff. You mean more cool stuff?
Additional conversation.
There's some more squid ahead of us coming across. So the pilots are pausing here for a minute. They're discussing troubleshooting. There's an ilex squid coming in from the right. They're definitely the most dominant biota I've seen. Definitely the most dominant macrobiota, at least. You can actually see more in the distance around noon. Yeah. Yeah, back moving. So we got more of these ilex squid. Looks like this sand dude's going to drop off to the left-hand side. Okay, so Ken's chiming in on that cutthroat eel we were looking at earlier, which is probably the same one that we're seeing here at 3 o'clock. And he says, overhanging snout, white lateral line pores, undulating worm-like, just above substrate equals Liliophis. And possibly Liliophis bruneus. Thank you for that.
Very cool.
Mm-hmm. We got another tripod fish, looks like. Yeah, so we're still making our way to the base of this wall. Ooh, here's another one. What's this guy here at 4 o'clock?
Snap zoom on him.
He's got a cool tail. Very ribbon-like. Very flat head. Almost looks like an alligator.
Oh, like a soar.
Heliosaur?
I don't know. Maybe.
Helosaur?
Halosaur?
Halosaur?
Yeah, halosaur.
Yeah.
Yep. I don't think we'd seen one of those on this expedition. I like it. This great big eyes looking up. You can see probably sits on the bottom looking for food above it.
Those giant black eyes. Really flat-faced. Ribbon tail. Really neat. I like how neat that is.
Oh. Thank you. That's great.
And then some dead tetrahedral sargassum.
So much to see, even in this relatively wide open, sandy landscape.
Barren area. Here's a different shaped bioturbated hole. Elongated. Yep, like detrital sargassum in there. So the Sargasso Sea is technically the Atlantic. In the Atlantic, right? But
sargassum is everywhere.
Yeah, sargassum definitely grows off in the Keys and the shallower water. Grows off the bottom, really tall, and then it breaks off and floats. And that actually becomes a habitat in and of itself on the surface. And you'll get like sargassum fish and stuff. They look actually like sargassum. Oh, nice. Oh, it's a head. It's a half-eaten head.
Squid has been here.
Munch.
That looks like one of those.
He left the brains.
Lophogastrid shrimps, I think.
Sea pen coming up at 9 o'clock. Yeah, sure.
Another tripod fish there at 2 o'clock, 3 o'clock.
Maybe the crinoid, too.
Oh, yeah.
So this looks like the similar species to what we saw earlier. Scott, feel free to chime in and tell me what you think this is. I can get it down to Penachalacia. That's about it. That's neat. So a few dives ago, we were actually picking up rock pens, which are a pretty rare form of sea pen. This one actually grows in the sediment like most sea pens.
But some seem to have figured out a way to live on rocks.
That's neat. So it looks like 4 polyps per row. At the base of the polyps, like these little pink dots. Not sure. That one has 5 polyps. 5 polyps per row as you get deeper. That one has 6 polyps per row. So they seem to increase the number of polyps or decrease as they grow up. Right? You can't see the peduncle. That would be in the sediment. That's pretty cool.
Very cool.
We're good whenever you guys are.
So I was completely wrong on halosaur feeding when I was musing there.
Oh, yeah?
Yeah. I was talking about their large eyes, and maybe they're looking up for their food, but Ken straightens me out and lets me know that they're bottom feeders and they actually wind up ingesting a lot of sediment.
So that's more for watching for predators, probably. Having those big giant eyes on the top of his head.
Yeah.
Another crinoid here. Right, went off screen. Looks like our sand dunes piling up in front of us. Another little mound to go over.
A squid was at that spot, so it ran off.
What is that? A fish. Yeah, we also have something in front of it at 6 o'clock. Maybe nothing, though. Yeah. Ooh, and a sea star. All right, we got a fish there. Little rat tail guy. And then if you go full wide at 9 o'clock, there's a sea star. I'm sorry, 3 o'clock, there's a sea star. My dyslexia is really bad today. Sorry, everybody. Yep. Cool. Definitely Goni asterid. Very thin arms.
Thin arms, but a broad central area.
Mm-hmm. Nice. He's about 15 centimeters wide. Thank you. Hey.
Ahoy.
Hey.
Ahoy.
Chris, how are you?
Good. And a happy Monday afternoon to you.
Yeah.
So the interesting Goni asterid that you just imaged, that's called Nymphaster arenatus. And it's actually one of the most, at least by dredging records, it's one of the most commonly encountered Goni asterid sea stars in the Gulf of Mexico.
Nice.
But given how many specimens we have of it, it's actually not really well understood, like its ecology.
Yeah.
And one of the things that I think was like a real, to me anyway, a real revelation was the fact that when we were seeing it, you see, if you remember how the disc is kind of sunken, and also the arms kind of flatten out. So I think what it does is it takes kind of a snow paddle approach to its body form, and because it lives on these soft bottoms, and it feeds on a lot of the things that live in the sediment. So it kind of flattens out and takes advantage of the fact that its body is sort of flattened like a snowshoe. These are actually sea stars that occur in the Cretaceous chalks of Europe. So it's a fairly old, or thought to be a fairly old animal. And I think during the last paper that I wrote for the Okeanos starfish stuff, we also saw a little yellow polynoid worm that lives in association with it. So there's possibly whenever we see one, there's possibly some kind of clam or some kind of other animal right below it. So perhaps the next time we come upon one, it might be interesting if the thrusters from the D2 blow away some of the sediment. But that said, it's probably not going to be terribly clear either way, but it's probably feeding on something. I think I probably overinterpret sea stars feeding whenever I see them, but that's what they're doing a lot. So, anyway.
Can you type his name into the chat for me?
I think he said Nymphaster.
Yeah, no problem.
Yeah, but I'm a terrible speller.
So nymph like the nymph, like like
Nymphaster.
yeah, Aster, yeah. Arenatus, Arenatus. I'll spell it out for you.
Thank you.
I think we've
chat.
got that one, Chris.
Yeah, Infestor.
Thank you.
I
Bye-bye.
found it.
Thanks.
Yeah, so if I don't get the first 3 letters right, because it does a lookup into
Right.
the taxonomy that exists from World Register of Marine Species. So it's actually automatic our logging system. So I'm talking about CTube right now. Our logging system, I go and type the first 3 letters of any critter's name, scientific name, and it pulls up all the data. So if I spell it NIPH, it's just I can't find it at all.
Right.
So I have to know how to spell it a little bit.
There's that tripod fish again. It's quite common here.
Yeah, something up there. A little fish of some kind.
Sort of eel-like.
In the far distance.
The direction we're headed, though, I think. Maybe.
Oh, what is that?
Oh,
Something
yeah.
in the
Now
water
we got
column.
a little jelly here. Kind of looks like Paralia from this far.
Oh, cool.
Yep, it is.
Now, is that red from like if we got close, would it be red?
Yeah, it's red. It's a nice dark red. This is a Scyphomedusa, so a lot of the medusae we've been seeing are hydrozoans other than the paraphila, but this is a not too distant relative of the moon jelly.
What's the genus, you think?
Paralia.
Paralia.
Yeah. We've got George Matsumoto in the chat room, so he can always... Oh, no, he's left, so it's up to me. But I'm pretty confident about that one. See this one from time to time.
You can see its tentacles. I'm not sure if it's coming through on the internet, but they're very fine.
Very fine. You can see those little indentations. That's where it has its sensory structures. More I look at this.
Can we get closer to it so the color pops a little bit? Is that okay? There you go. It's getting a little brighter.
He might give us a little look on his underside.
Don't make this awkward, Alan.
Thanks for that. I was thinking the pilots. Yep.
Yeah, we've been on this boat a really long time. Sorry. That's neat.
It is really neat. It's beautiful.
There you go. So, that's his mouth, right?
Mouth with its dangling oral arms. So its mouth was in the central area of those oral arms that came out there.
And I would assume a lot of these long tentacles are probably a stinging cells.
That they
See if
will
they're
have
needy?
stinging cells. They're nitey in the tentacles.
Uh-oh.
Yep.
Sorry, guy.
Got a little tipped upside down, but now we get a great look at its underside. You can see the generally 4-part symmetry of its mouth region there. These nice radial canals. In that central area, you can see through the top there, that's where its gut is. And then it distributes nutrients.
Look at the texture on it.
Yeah.
Look how neat that is. So what's the point of the texture? Is there some kind of purpose for that?
I don't know. That's a good question. But this is kind of a cruiser that'll be, filtering fine sediments. It's actually paddling. So a lot of times we think of jellies as having jet propulsion, but this is like a big round oar or paddle. And so its movement is pushing the water the same way a paddle does. Really nice. Really
Beautiful.
nice.
That's awesome.
Thanks a lot.
I'm glad we were able to get close to it. From far away, you can't see the color. It just looks like it just disappears, right, in that water column, that red color.
And a lot of times for the jellies and a lot of other organisms, we want to see them from different different orientations. So we got all the orientations on that one.
I know. Doing flips.
Yeah.
You got some more cutthroat eels maybe at 9 o'clock.
They're still below 970 meters.
Yeah, we're shooting for 930.
Uh-huh. Great. We'll keep going and we'll see what we see. Huh. So Ken Sulak is in the chat commenting that the tripod fish that we've seen is relatively common, but hasn't been imaged in situ too often. And so especially the view we got of it sort of just offset from head on is one of the best to his knowledge. So that's
Awesome.
Really nice. It goes on to comment that most tripods are really up high, but this one sometimes has been seen sitting right on the bottom, right on the substrate with the long pectorals folded against the body, trailing back to the caudal fin. This might be facultative feeding mode, targeting, benthic, deeper benthic inverts. Very interesting. Thanks for that, Ken.
What's that? Maybe a fish? Yeah, it looks similar to the one we saw earlier, the little short one with the black spot on its dorsal fin. Kind of shiny. I forget its name.
Polymyxia.
That's it.
I don't know. There's a certain element of the, creeping across the sand that's like the midwater dives. That's sort of, it's not always full of stuff, but there's sort of a constant, steady stream of interesting things to look at.
So not a lot of biological density.
Right. Although there's probably a lot going on in the sand there.
Here's another jelly.
Oh, another
maybe?
jelly.
At noon.
Might be the same guy. Sort of has that same look about it from here.
Yeah. Looks like a bowling ball.
Yep.
Really round.
Little Paralia. Yeah. Definitely. To my knowledge, reasonably well-known group of jellies.
really cool looking.
Just so... Just watch that motion.
Mm-hmm. Super chill. Bloop.
Yep.
Bloop.
Great. We'll keep our moving. That was a really nice video of that animal.
Mm-hmm. So what's his solitary life stage? Does he have a solitary? I mean, not solitary. Sessile life stage.
Yeah, I'm almost certain that one does have a benthic stage. Ooh, what is that?
It's the same one, isn't it?
Oh, yeah, it's just the jelly.
This is a jelly through a wall
I
of
saw
schmutz.
something behind. Felt like I saw like a column behind it. It's nice. Got the size on that jelly. Makes me curious about Paralia polyps. Don't know if I've ever seen them.
Now the cutthroat eel likely at 6.
There are jellies that skip the polyp stage. Actually, quite a lot of the ones that are open ocean, they just skip the polyp stage. And then there are, as I mentioned, a whole bunch in the jelly group that skip the medusa stage. And
isn't there one that's supposed to be, in theory, like immortal? Was I reading that?
yeah, there is more than one. There are several jellies where the medusa stage has been shown to be able to sort of de-differentiate and then turn into the polyp stage. And so that's why they call it immortal.
Okay.
But when you think about it, a lot of asexual organisms that have asexual reproduction, that
it's kind of the same.
Yeah.
Sorta.
They're also kind of immortal.
Another squid here coming up.
But the one you're referring to is called Turritopsis nutricula. And it's particularly well known as being the immortal jellyfish.
Right.
But there are other jellyfish that can actually, it's been shown, can do the same thing. Yeah.
And I remember in Australia, they have irukandji. Is that it? In Australia? That's a box jelly, though.
Mm-hmm.
It's a really nasty sting.
Yes, that
it's
is
like
a
deadly
nasty
for
sting.
humans, isn't it? It can be deadly, right?
Irukandji, not usually, but occasionally. But it also can debilitate people. So if swimmers get irukandji, they could be trouble.
Yeah. So that's a little bioturbated pit with some detrital sargassum in it.
No, I think so. I was wrong. Paralia is a direct developer, which means that it skips the polyp stage, which is consistent with, it's sort of...
Why am I not seeing sessile jellyfish when I'm out doing our... I've never seen one in my mind. I don't know what they look like.
You mean the polyp stage?
Yeah.
Or the... Oh.
Yeah.
They tend to be very small, yeah. So
I'd only really see it if we were like super zoomed in and really focused on it.
By and large, yeah.
Okay.
Millimeter, 2 millimeters.
Oh, tiny. Okay.
Yeah, we're talking tiny.
Alan, I'd like to ask a question about along the same line, and that is there's obviously a challenge with determining the life cycle for something that lives in the deep sea or lives way offshore in open water. So how do people go about determining that X is the polyp stage of Y and vice versa?
Well, in years past, the best way to do it was to bring the medusae into the lab and then get them to spawn, and then see what happens after the eggs and the sperm meet and an embryo develops. Does it turn into a polyp, and if so, then what type of polyp? And that sort of study is continuing to go on now from time to time. But then we also now have genetic tools, which have helped us out a lot in matching up polyp stages with medusa stages.
Is it really difficult to get jellyfish to breed in captivity?
Some yes, some no. So there's, because people like to see them on display so much, there's quite a lot of work to
Sure, the aquarium industry.
husbandry. Yes, exactly. And but things like the moon jellyfish or Cassiopeia, the upside down jellyfish, these are really quite hardy animals and easy to keep in captivity. And you can raise all the way through their life cycles. So. That makes them good study organisms. Cassiopeia, in fact, is a widely used study organism.
They are the coolest jellyfish ever, because you're out snorkeling and you see them upside down. It's literally an upside down jellyfish. Like, it lives with its bell facing the dirt, and it's got like zooxanthellae in it, doesn't it? Is it the same species that's in coral, or is it's the same?
Different species.
Different species, but same dinoflagellate thing.
Yep.
Yeah. And they have these like little paddles, and they're like beautiful blue colors.
Yeah. Blue and green.
Yeah, they're gorgeous. However, do not pick one up because it will sting you. I know that from experience.
It can sting you even if you don't pick them up.
Yes, that's true. You should not touch nothing in the ocean. If you don't know what it is, don't touch it. Even if you know what it is, you probably know better than to touch it.
But these upside downs, they actually produce a stinging snot.
Yeah.
Ooh, there we have something to look at.
Oh, a crab.
Nice crab.
Red crab. So that's Chaceon.
How do you spell Chaceon?
Chaceon? Yeah. And it's Qui quincadens. The red crab. There you go.
Got it.
John and I did a biogeographical paper on its cousin, Chaceon fenneri, off the east coast of Florida. That was my first paper I did when I started at Harbor Branch.
Mm-hmm.
I sat there and measured all their carapaces from old footage.
That's nice.
Yeah, it was fun. My calipers.
Run away.
You can see how soft the sediment is when he steps in it. It's very silty.
Mm-hmm. So we're still at 973 meters. Oh, no, it's all right.
You don't... D2 doesn't have a turbo button? Oh, okay.
It's all good.
Go as fast as your thrusters can carry you. Such is life. Now, if we could flip this crab upside down and look at his undersides, we'd be able to tell if it was male or female, based on the shape of the.
We'd be looking for the Washington Monument, wouldn't we?
That's right.
You know that from eating our blue crab.
Mm-hmm. They all have that same kind of. I forget what that's called. Plastron? Is that it? The plastron, the underside? Going back to high school, college.
Invert
bio,
zoo.
Yeah. I think that's right.
Telson?
Well, I just have to have stepped out of an invert zoo lecture if you want some more.
Plastron, right? That's the underside of the crabs.
I think you're referring to the abdomen, which is greatly reduced and folded up underneath the thorax.
Yes, abdomen. Okay.
Yeah.
Plastron is a sea turtle. That's where I'm getting that word. same thing on a sea turtle. Yep.
Ooh.
So that's a squat lobster.
That's a nice looking squat
He's
lobster.
got a really long antenna.
Kind of want to scoop him up.
Good luck, man.
This is really good timing, actually, because the squat lobster is kind of intermediate between
That's
the
right.
lobster and the crab we were just discussing, and you can see the abdomen there kind of curled under a little bit.
Yeah.
Do you think
And
you
if
could
the other
collect
crab
that?
would be so reduced and curled, it would be held up against basically the belly, the ventral margin of the thorax.
Right, it's like it's not quite sealed in there, but it looks like it's close to sealed. That's where the females will keep their egg sacs too. So, if you're out crabbing, and they have that,
Oh, he's pretty big.
yeah.
Yeah. Let's just take a good look at him. Thanks.
So the ones we were seeing the other day were stark white. This one has some red on them.
Yeah, it's quite an interesting, different look to it, doesn't it? That's.
Yes, he's a little hair short of 10 centimeters.
Yeah, let's avoid it. Thank you. This will be some good footage that's valuable. Look, wow,
Oh, look at his
eyes.
That's so neat. It's like cloudy, compound eye.
See his
Look at
rear
this.
leg broken?
Look at that
Oh,
brittle star next
There's
to
a
him.
really tiny brittle star. It's got a cap on it.
Yeah, he does. That's neat.
Okay, to me, that's weird. I don't know if that's weird, but that.
Yeah, it looks weird. It's totally weird. It's totally weird to me, too.
The other thing you can see on this crab, it only appears to have 4 pairs of legs. It is a decapod. It does have 5 pairs, but that 5th pair are very reduced and kind of tucked under that curled abdomen.
I think a lot of them you end up using those back pairs of legs to hang on to gorgonians. This one's
Thanks.
a ground
Video.
dweller, so
Great.
he doesn't need them for that.
And that brittle stars just cruised on by, huh?
Yep. I don't even know where he is anymore. Yeah, teeny thing.
Ophiotholia, Chris Ma says. Oh, he wants to watch it if it's possible to find the brittle star.
Can we go back to that, guys? Or not?
It was out front of the crab, the Galatheid.
Hi.
Hey, I know we've pulled away, but has it completely gone out of visual range?
We're going in and we're
We're
zooming back
seeing
in
if we
right
can find
now.
it.
Okay.
So it's going to be 6 o'clock of the... Yep, go ahead.
I'm sorry, but I mean, there's a whole bunch of these weird brittle stars that show up in the books all the time, and they have these bizarre discs and shapes, and nobody knows what they do with them. And I think on the Okeanos dives, I've now seen at least 2 of them. This is like the first time I think I've ever seen. There's this one that has a disc that looks like a Hershey's Kiss.
Yeah.
Yeah, that's what this looked
raised
like.
up. Yes, and there are a couple of genera. I forget exactly, but the genus that comes to mind is Ophiotholia, and I think one of them has like spines around the disc or something. But the fact that you just saw one alive just literally made my heart stop, because I don't know if anyone's ever seen one alive before. And it was the same thing like when we saw the brittle stars out at the Astroophiuras alive when we were out on Johnson Atoll. So I'm really curious to see if we can capture that video. I mean, it's almost more important to see it alive and doing something than it is to actually like collect it. So I'm just really, that would just be really amazing if we saw it again. So I mean, I know there's like that few brief seconds of it moving across the bottom, but
we're having trouble relocating it.
Well, it was right next to the squat lobster, right?
Yeah, but it was on the move, and somehow, yeah, we've lost it. But we'll keep our eyes open.
You know, I noticed some white thready-like stuff just ahead of the squat lobster, and I wonder, Chris, would these submerge themselves into the sediment? Is it possible it did that? And
It's possible, yes.
picking up.
Again, it's possible. Nobody knows what they do alive, so it's possible it just sort of... I mean, some people have speculated that they bury themselves and that the cone is what sticks out of the top. But again, it's one of those things where...
Could that be it?
You know, it's like extrapolating extraterrestrials. We don't...
Now, just to the left? And... yeah. No.
Chris, I think we lost it.
Yep, we've lost it, sadly.
Sorry.
Okay. No, that's fine. I mean...
I was able to see it pretty clearly when we were looking at the lobster.
The
So
video
I
should
know
be
we got
pretty good.
some of it. It's just kind of off to the side, and we weren't focused on it. So I don't either. That is the fastest ophiuroid I've ever seen in my life, or he went down into the mud.
That's
Not
possible.
really sure.
I mean, that's fine. This is kind of the fun part of this, is seeing these bizarre, like, you know, I mean, and they're almost mythic in the way that they, you know, you just saw one for a minute and then it disappears and you don't see one again for like 5 years.
Pegasus of ophiuroids.
But at least the video is there.
Yeah, at least the video is there.
Yeah. I said it's like the Pegasus
so
of
I
ophiuroids.
can... Yeah.
Oh, let's... We got it. Squid eating
Yes, okay.
crab.
Oh my
goodness. Yeah. Squid eating crab.
My squid.
That is very nice. All right, I'll let you guys... I'm sorry, that was just... I'll
Awesome.
have to go back and check the video.
Yeah,
Thanks
sorry.
So much.
We'll look for another one, okay?
Bye-bye. Yeah, no worries. Thanks. Bye-bye.
So that's a red crab eating a big old squid. Another squid here at 6 o'clock. It looks like he might be eating or something. Oh, no. Okay, so he's facing out of the hole. So his tail end was in the hole. Okay. Yeah, it's super flat. Like we haven't even hardly changed depth. If anything, we've gotten deeper. Are we going the right direction? Like, where did we land? It looks like...
Well, you know, we're here and we're seeing interesting things, so.
That's the nature of exploration. You never know what you're going to find.
Exactly.
Just have a good hypothesis.
Another squid here at 3.
Hanging out on the seafloor.
Yep. Kind of like us.
Yeah. But you know, that last zoom at the at that crab, this shows how much we're sort of going over that we're not able to see, right?
Yeah.
There's many small things that we tend to notice only once we zoom in on something
That's right.
larger. I keep thinking about that isopod that we followed as it drifted down to the bottom, and as soon as it hit the bottom.
So I'm reviewing that footage.
It went in
next to me with Emily. She's our video clipper right now, and I can see the ophiuroid like right in front of us.
The Ophiotholia?
Yeah, like I can see it. I just didn't know where to look. That's okay.
Oh, are we back? We're not.
Different one.
This is a different one.
Same species, maybe. If we get a snap zoom on that guy,
Maybe another Ophiotholia is there.
that'd be awesome. We won't see it unless he moves. They just disappear.
No, this one's smaller.
So that one
Very
has
nice.
a big, look at the spine on its back.
Ooh, wow.
Did we see that in the other one? Almost like a rhinoceros, right?
Mm-hmm.
Triceratops.
You can see the reduced leg there and the tucking in the back.
Mm-hmm.
It's prehistoric looking, right?
Yeah, right. Armored.
So this one, is this also too large to be an easy collect? Yeah, I think so. If it's relatively easy to get, then yes. Oh, if the whole ship has to move, maybe. Hello, squid. Maybe we should leave it. Maybe we'll come across another one similar sized. Okay. Yeah, absolutely. Thank you.
Do we have a better name for this than squat lobster? Did anyone chime in
No
on
one in
that?
the chat room has put in there. Scott France does comment that it may dart away.
Is this sample 4 now?
This is sample 4. We'll see if maybe this will make a nice, interesting collection for Galathioid worker like Martha. Scott France says this is Munidopsis tridentata. The three-toothed squat lobster.
I think it had 4. It looked like it had 4 spines.
4 spines? Maybe
Yeah.
it's quadradentata.
Yeah. He might just run off. Let's see what he does.
Let's see what he does. I think he's going in the chute.
So he's going to swim backwards. So maybe the best way to come at him is from the backside. So when he jumps, it goes into your tube. Yep, there it goes. He was able to swing his rear end around and go the opposite direction though, so it's pretty quick. It's up to you, Alan.
Yeah, why not give it a little shot? We're here. We've got.
I'd say one more and then we go.
That's good.
Fair?
That's of course.
Is that fair? I don't know what happened to the wall. Like, where is this wall?
Don't know.
I think you're gonna have to come at him really quick. Like full blast suction and just like.
Scott says that you had good advice and that they're very hard to collect if they're not holding on to something.
Mm-hmm.
So we'll just see what happens, and then I'm confident. Confident.
No!
Oh,
Bye.
you little
Bugger. That's okay. You can give it up.
But thank you very much for trying. That squid is hanging out. It looks like it might be interested.
Yeah, go eat him. Bopping around. You can eat him. Go ahead.
Then we can watch.
Go get him. Oh.
He is looking. Huh.
Oh, come on. Right behind. Right behind the suction hose. And he's gone. Oh, he's still there.
He's still there.
The squid is hunting him.
I don't know. Maybe not.
Maybe he's more distracted by us.
Yep. It's going away.
Yeah.
Yes,
Speed
please.
up. Hit that turbo button.
Probably most of the Scott's making the comment that a lot of the mobile fauna out here on the soft sediments are probably collected from trawling.
Mm-hmm. Some silvery fish, maybe. Another squid.
Yay!
Oh, nice. 80 meters from what appears to be a wall on the sonar? Or maybe something? More of these school of ilex coming through. This is the squids we've been seeing.
Yeah. Again, looking so much like a patrol.
Mm-hmm. I'm going to do a water sample at the base of the quote-unquote wall when we get to it. Does that work, Alan?
Yeah, absolutely. Wow, so many.
I know, right?
Yeah.
Way off in the distance, right by the end of our light pool. That's what it looks like. They're just hanging out, swimming.
Yeah.
Get too
Mike's
close.
curious if ilex will be in the eDNA. Somehow I think so.
Yeah.
Well, we'll be taking another sample soon.
Shrimp or something?
Nice.
Water column shrimper?
Mm-hmm.
He's very skinny.
Very skinny.
Different than the other
very
ones.
long antennae. Yeah.
Might be a sea pen there at 7 o'clock. Yeah. Looks like similar species to the one we've been seeing.
Yeah, I think it is.
It's pretty small. The other one had at least 5 or 6 pops at the bottom. At the bottom first row. This one looks like it's only about 2. Maybe 3. And there's some sargassum, detrital sargassum behind it.
Trying to convince myself that I saw nodes in that thing and that it would be a bamboo coral.
Wishful thinking.
Yeah, it is wishful thinking.
We're good whenever you guys are. Thank you.
Sure looks like something like nodes.
Nice.
Maybe it's just curled polyps.
It looks like an active burrow here at 9 o'clock. Freshly dug.
Nice volcano.
Mm-hmm. There's something dark at 3 on the sediment. It looks like it could be a fish, maybe, based on the movement of the one end. Yup.
This will be nice for the fish folks.
Some type of rat-tailed fish.
Some sort of cusk eel. It's all right.
Yeah, I'm gonna just keep moving. No big
Yep.
deal.
See if we can get to that. That's something. Don't want to call it a wall.
Yeah, not yet. So it looks like we're about 45 meters away from something on the sonar. Yeah. Not a wall.
It's all right. Whatever it is, we'll go check it out.
Pile of gold doubloons.
Yeah, it's the treasure we've been looking for.
Quick, cut
What?
the feed.
I love this ilex.
Yeah, they're neat, aren't they?
They really are.
Little bit of a rock, maybe.
Well, if you have a little rock outcrop, that'll
Have something on it.
get some substrate for something for sure.
I don't know how we haven't found this like rise yet. Not sure what's going on. Unless the multi-beam is insanely off, which doesn't make sense.
Yeah, somehow I don't think so, but who knows?
We have a place in in the North Florida Marine Protected Area that the multi-beam that's a couple years old shows, hard bottom, really obvious hard bottom, and then we'll go back to the same spot. Sometimes it's totally filled in with sand, and then we go back the next year and it's cleared out. So it could just be sand movement, but it seems like a lot of 180 meters of sand is a lot of sand.
Right.
So I don't know if that's the case here, but that does happen on the shallower or less rugose areas in the North Florida and PA. I've seen.
I can't remember the exact date, but I think that mapping lead was mentioning that this was mapped a number of years ago, so not just, very recently. Yeah,
Got something coming up.
it's like a rock. Also another Larvacean house going by. Or old Larvacean house.
There's another little fish and some tripod fish. So there's actually two species of fish here. There's one at like 3 and one at like 4 or 5. And then another tripod fish off the screen, just off the screen. But most of this is still just bioturbated sediment, and a lot of these dark spots are detrital sargassum, which is a type of brown algae that floats down from the surface. And then the ilex squids. This looks like a different fish. Have we seen this one before?
I don't think so.
He's neat looking. Again, most of them have these barbels right on the bottom. I'll let them feel around in the darkness. Maybe some worm poo behind his head. Actually, there's some in front of him. Let's see the pile of poo in the back.
Mm-hmm.
He's neat.
Yeah.
Ooh, hi, squid. Excuse me.
Still making our way.
Looks like something just off screen there. The shadow of somebody. Maybe a squid. Squid shadow. Squid shadow, squid shadow. Okay.
Sounds great. Huh. Wonderful.
Weird. This is like, maybe it's just a sandy slope now, but we haven't really come up.
Well, maybe whatever it was moved, slumped.
That fast?
Since it was mapped. Obviously, I don't know.
What's that yellow thing? That's dead center.
Oh, yeah, let's look at that.
Oh, squid ink.
Squid ink, yep.
Lots of critters here.
Yeah.
That's promising for having a hard bottom nearby. Fish usually have a tendency to conglomerate around rock bottom. And we're seeing is like white on like consolidated clay-like rocks that we have collected before. This is the extent of the hard bottom in our sonar.
Well, it is what it is.
Look, a rock.
It looked like.
Lots of squid in front of us.
Yeah, they seem to be, just on the edge of the lights a lot. Well, we have had dives where there were no fish.
It's true. We did off the Lake Spur. There was like no fish out there.
Yeah.
It's a little bit of a slope here. There's something. I think. Might just be a rock. I think so. Okay. Never mind.
It's definitely more slope here.
Yeah, you can see the increase in slope in front of us.
But still big, largely bioturbated
Yep.
sand.
Is that it? Can we move forward?
Oh,
Okay. Am I seeing a wall? Maybe? Maybe?
maybe a little outcropping of some sort. Yeah.
Yeah, it looks like layered rock.
Nice.
Yeah, found something. Get closer to it. We'll
If we
figure
could
it out.
snap a water sample while we're waiting,
Yeah, we
this
could do
would
that.
be a great spot for it.
This is going to be 4W.
Yeah. And this is Niskin 3, correct?
Yes.
Yeah, would have to be.
Thank you.
Thank you very much.
Yeah, so now we're at the base of this wall or some kind of rock formation. It's like a layered slope.
We know where we're going to spend the... Ooh. Can we zoom in on the organism floating in the by? Yeah, probably not enough time. All right, that's all right.
Hazards.
Great, thanks.
So I don't see any really large macrofauna at the moment. Maybe a blackfish here at, 6, 7 o'clock? And then it looks really heavily sedimented on top, at least locally.
It's pretty big.
Yeah.
Wow.
Well, it looks like in the sonar it actually ends.
That's...
So it looks isolated, but let's see. Yeah. Are you going to go over it? Okay.
Yeah.
Sure.
You don't explore if you know what you're going to find.
All right. Then it's not technically exploring.
Exactly.
Yeah, so it looks like the base of it is that like consolidated clay almost, like we were seeing earlier.
Mm-hmm.
That's probably soft. Not really soft, but like a softer rock, and then on top of it's very sedimented. Still have some bioturbated sediment on top.
It really does look like you could see it going,
Yeah.
falling slumping all at once.
Yeah, like an avalanche or something.
Mm-hmm. It doesn't look stable.
No.
Oh, there's an interesting plankton going by.
Just out of focus there. Ooh. What is that?
How's that flappy thing again?
Yeah.
I don't know if that was a pteropod.
It looks like we've got some areas too where something has slid down this. Looks like sled marks.
Mm-hmm. Piece of sort of rock, or
Not quite yet a rock.
yeah,
Squishy.
I can see some sediment there. Wouldn't it be interesting to see the whole thing slump?
I don't think the ROV team would be very happy with it.
I didn't say it.
I feel like it would just get cloudy. I don't know if we'd see anything. Yep, looks like a gorgonia or something at 9. Yeah. Yeah,
Yeah.
sounds good to us.
Star.
Ooh. Where's the star? Off the right?
Mm-hmm.
I missed it. Hi. Chris, is that you?
Chris, is that you?
I can hear now.
Yeah. What is that? 3. Can we zoom in on
something
the
at
rock
3?
there at 3? I think it's one of those lobsters.
Oh, makes sense.
By the way, right. Yeah, the right at 3 o'clock.
I think those might have been whole thurians swimming by.
Hey.
Yes. Is that
Mike?
This is Mike Vecchione.
Hey, Mike.
I just wanted to let you know the flappy thing you were asking about was a holothurian.
okay?
Nice.
Thank you, Mike. I wondered if.
Okay.
Thank you.
Bye.
Bye. This looks like an armored shrimp of some kind.
See a couple small demo sponges there. They're sort of bluish to the left of the squid.
Mm-hmm. Yeah, we're good. There is a Nidarian, maybe. I'm not sure.
Ooh, we see sediment coming down.
Yeah, look at that.
Yeah.
Little river sediment.
Yeah, look how unstable it is. Sediment shoot, says Kevin Rademacher.
You can see it moving, yeah. Real slow, but definitely there.
Whoop.
So that's a pretty big slope right there.
Yep. So it really is looking more and more like this what we expected to climb all had slumped away between when it was mapped and when we got here.
Yeah. So I think there was chatter about remapping this.
Yeah, after the dive. I think that's really, really interesting.
Yeah, quickly
And it could
sediment
be
can move around. And I don't know if it was a wall.
I mean, we should
It
be
looked like
at
one.
Yeah,
We should be at
830 meters, and we're really at 963. So that's 130 meters that, if everything was correct on the mapping,
Yeah.
that is gone.
Hey, Alan, what year was it mapped?
Do you know the year? I can find out, John. Hang on.
But you can also just see how unstable it is. Get a snap view in on that eel if we have a chance. Yeah. Oh, there is the halosaur again.
So,
Nice.
John, Derek, who's our mapping lead on the ship, said he thinks it was 2014.
Okay.
Yeah, so it's 10 years or so.
Mm-hmm.
Uh-huh.
But we've seen that before, where the water, you see the sedimentation move, like when we're on the MPA cruises over the summer. We do that once a year, and the sediment moves a lot. And it really, sometimes like one year it's all this gorgeous blocky rock, and the next year it's just totally filled in. Same spot.
Interesting.
Yeah.
Really interesting. Thanks for that. Yep, all set. So this Tortugas Valley
Yeah.
could be very,
Yeah,
very
different.
dynamic.
Mm-hmm. What is going on there? That looks like a dead piece of seagrass. Thalassia, maybe. It's just standing up. Yeah, look at this like river of sediment.
Yeah. Yeah, you can see why there's just not much opportunity for benthic colonial organisms to get a foothold.
Oh yeah, they just get wiped out. Slide down. There's nothing.
Buried.
There's nothing to settle out on. Sometimes coral will settle out and then not find suitable habitat and then jump back up into the water column and try somewhere else. So, or their larvae will do that. There's really nothing for them to grow on.
We got a colony there.
Yep, looks
There's
like
a little
Chrysogorgia.
life holding on.
Yep. Yep. Chrysogorgia.
Trying to something. There's sponges on the wall in the background. You can see their oscula really nicely. And another colony to the 10 o'clock there.
And 2 squat lobsters in there. It's also, did you mention the sponges?
Yeah, sponges
Oh yeah,
all around.
There's
yeah,
some blues and yellows on the side too.
blues and yellows, and
That funky looking one at the top. There's a shrimp here at the base at 6. We're good whenever you guys are. Oh, maybe an anemone? Right,
Oh,
right
yeah,
above
just-
laser right now. If you snap zoom, there's another anemone, but it's okay. You can keep going. Yeah, it's right there. That guy. Maybe a solitary cup coral right below laser. There you go. That looks like a cup coral to me.
Yeah, it's nice. You can see the skeleton through the tissues of the cup coral. I'm used to seeing those skeletons in the collections at the National Museum.
That's how you identify them. There's actually, like, I don't even know, hundreds of species of cup corals. And the only way you can really identify them is to take that flesh off and then look at the heights and the sizes of those. Well, the corallite, which is the actual place the anemone grows out of. So those little, like, ridges that are in there, and whether or not the third one is taller than the seventh one, and like all these crazy things you have to do to get the IDs on them. So we just go with cup coral.
Yeah, but did you know that I think that solitary corals are there are more species of them than even the colonial kind, or pretty similar in number.
I wouldn't be surprised.
Yeah.
They're and we got them in the Arctic too. You know, like they're in every ocean.
Little tiny ones and big ones.
That one was pretty big.
Yeah.
Yeah, actually, there's quite a few more of the solitaries than the colonials.
Yeah.
Steve Cairns did his, I think, his dissertation on those, and it's an incredible number. I don't know how many, 500, or I can't guess, but over 500 species.
Yeah, I can't even.
Large number.
Yeah, so sometimes you can get them down to like family. Like, so Scleractinia is order, and then sometimes you can get them down to family, but for the most part, I can't even go there. Like, it's a cup coral. You know, we'll move on. Oh, there's a fish at noon. Or a shrimp. I don't know what that is. Making a cloud. We probably blinded the poor thing.
Do wonder if it's...
There you go.
What's...
Well,
oh,
he's
it's
very shiny. What do you think it is?
I don't know.
Aww. I think we blinded him and he shot himself into the dirt. Maybe a midwater fish? It's very shiny for a...
Myctophid. So, Myctophid, but not cyclothony. So it says Mike Vecchione. So yeah, midwater fish that's confused and got a mouthful of sand.
Oh, poor guy. Sorry.
It's a really good view of it, though. Oh, there goes Baroe floating by. That's a Tina 4. That's an interesting looking Myctophid.
Thank you.
Great. So we'll have a little pilot change here, and then about 30 more minutes at tops of bottom time, right?
Are we out at 3 o'clock? We getting out at 3?
Not sure.
Can you, by looking at the slope sideways, or parallel with the slope, can you determine the angle of the slope?
I'll ask them, John, as soon as they're done changing pilots, I'll get them to look sideways for me. It'll be a few minutes, though. They brief each other before they switch seats. So they're briefing each other right now before they do their turnout.
Have you seen any actual vertical part of the wall, or there was none?
There was one little section that was maybe, a meter and a half tall that were like now on the slope above it. So yeah, there really was no wall. And according to the multi-beam and where the boat and the ROV is on the high pack, it looks like we should have been at 800 and something meters by now.
Yeah, we're above 830. Yeah,
830 meters, and we're sitting at 960. So the thought process was this actually slumped down between the time the multi-beam was picked up and now. So there's rumor that tonight we're going to run some multi-beam lines over this and see the difference between what the old multi-beam says and what we're looking at today. So yeah, it's nice that we have the technology on the ship to be able to do that, and the staff.
That's pretty weird. I remember I had to leave around noon, but just before that, you were starting to get like these big mounds of sediment and exposed rock. And that was part of the stuff that slumped down.
Yeah, that's pretty much how all day has been, that like just kind of smooth or bioturbated sand with a couple of rocks here and there, but really limited. And then we just saw that one quick, like little mini 2 meter tall wall. And then we're kind of we've passed that now, like we've gone on top of that. So I'm not sure if or when we're ever going to hit any real shallower, 180 meter shallower area. We may not even get to it.
Makes me wonder how the benthic life responds to a large slump.
Yeah, right. Just smother it all like an avalanche.
Or can they get themselves out of it?
Yeah. Well, that one Chrysogorgid lived, or has grown since.
Yeah.
Oh, hey, pilots, are you settled down? Can you just turn the camera to the side so we can look at the angle of the slope here? Oh, there's something.
Yeah, we want to look at that too. Ooh.
Oh, it's at least 45 degrees. I mean, the way I look at this is I imagine a square on my television, and if it goes from the bottom corner to the top of that square, that's a 45-degree angle. So that's about a 45-degree angle. Yeah, go ahead. Yeah, just keep going upslope, but we have to stop and look at the octopus before
yes.
Yes, the Dumbo octopus.
We have to. Oh, it's so adorable.
Yeah, they're so cool. All right, get up and swim, buddy. Come on. So you can see his giant eye. Where's Mike Vecchione? Mike.
Serrate octopus.
Yep.
Opistoteuthus.
Opistoteuthus.
Yes. Dumbo.
He's cool.
He is so cool.
Like a little squeaker toy. Awesome.
I'm curious about its behavior sitting on the bottom like this.
I mean, they're pretty smart. I would imagine if something the size of D2 came at me underwater, I would probably squat down into the sand and sit.
I say,
It's
you don't
terrifying.
see me?
Yep.
You can see how fast it's like sitting in a snowstorm. It's, the sediment is it's accumulating on that.
Yeah. He has really
that octopod.
soft skin, too. Right? Looks so soft. Awesome. What, just hang out. Can you hang out for a second?
Okeanos.
Hey, Mike. Go ahead.
You guessed who it was. Yes, that's Opistoteuthus. So it's one name for it is a flapjack devilfish.
Nice.
Flapjack devilfish. Nice.
Yeah. And so they do sit down on the bottom. This genus spends most of its time on the bottom, but we'll get up and swim around. But they're benthic feeders, and right now it's just watching the sub, trying to figure out what the heck that shiny thing is up there. If you see the little sort of bumps that are below where the eye is, those are things that we don't know the function of. There's actually a progression of them that goes up each of the arms and up to the mantle.
And they're not photophores?
On this, no, they're not photophores. They are, I can't remember the name of it right off the top of my head, but they're clear areas in the skin. We don't really know the function, so I've been speculating that they are a way that the animal can gather extra light in and sort of channel it towards photoreceptors, because the layer right below the skin is really transparent, and I think it might function as a light guide for light coming in through these clear patches in the skin. It's a beautiful close-up of it.
Awesome. Yeah, look at his eye.
And I'm guessing, at first I thought it was Opisthotuthis agassizii, which is the most common one down here, but I'm guessing from this close-up that it might be another species, Opisthotuthis grimaldii.
Grimaldii.
Yeah, but I'm not absolutely certain which of those two it is.
That's
Okay,
all right.
Awesome.
Obviously, it's still keeping its fins tucked in and its arm tips all tucked in, so it's a little bit wary of this big mechanical thing that's hovering over it.
Yeah, 9 foot tall giant box with lights. Kind of scary.
Yeah.
Yeah.
Oop.
So do you have questions about it, or should I keep blathering on?
No,
That was super.
I just like
super
listening
great.
to you tell us stuff.
There goes a cloud
It's pretty
of,
cool.
sediment over it.
Yeah.
Okay.
Can you throw the lasers up
Yeah,
so I can see
that
the
was
size real quick? So yeah, he's about 10 centimeters wide. So he's pretty small.
Nice.
Little guy like this.
Mm-hmm.
Awesome. Thanks so much,
Yeah,
Mike,
they
for
don't
chiming
get
in.
very big.
No, yeah, he's a little guy.
That's
Thank
okay.
you. Thank you,
Yeah.
thank you.
And there's a squid in the background that just rammed into the bottom.
Yep. He's going to do it again.
He's not too happy.
Yeah, we're scaring him. Thank
Yeah, he's
you.
about to hit bottom again. Yeah.
Awesome. Thanks so
Wow.
much.
This is. There we go.
Not
Okay.
too bad.
Bye,
Not too
y'all.
bad, exactly.
Bye.
Thanks, Mike.
Opisthotuthis.
Opisthotuthis.
The flapjack devilfish. All right, we're going to keep making our way up this slope. Oh, here's another rocky area.
Yeah, we can check that out.
So this again looks like that wall we were seeing inside the sinkhole over on the far east of the Blake Plateau. It's like,
Yeah, I wonder if you can see the sediment on top of the layers that's accumulated and then sort of fallen below it. It's pretty
avalanche-y.
pretty clear. Yeah, it'll be interesting to see if anything has colonized the
It's like layered.
exposed part. Yeah.
I wonder, see the lighter area of rock here, it looks like it's been freshly exposed. I would imagine that before it slumped down, this was probably all covered.
Ooh, nice. That's a different tripod fish.
Cool. There's also some, like the blue sponge maybe in the wall. There is a ophiuroid just south of the laser, below the laser.
I think this is called Viridensis, the dark-headed tripod fish.
Bathyopterus.
Bathyterois.
Bathynomus PT.
But we should let one of the fishy people confirm.
Yeah.
But anyway, that's the picture it looks like to me.
Very densus. Cool. Oh yeah, look, he's got a big isopod on his underside.
He sure does.
Attached to his fin, maybe? Still has his smile on.
It's interesting that in that head region, it looks, it's transparent.
Mm-hmm.
Sitting there waiting for food. Is that another parasite?
It might be.
Or an amphipod that's
Yeah.
crawled up on him? I think that's good.
I think that is an isopod, amphipod.
Amphipod sitting
Yeah.
on its.
Mm-hmm. And the isopod down underneath. And then looks at another one below that.
Oh, wow.
Yeah, it's very parasite-infested guy. What is that? Can we zoom on that? Dead center screen? Sorry, right in between lasers. What is going on there? Oh, some kind of sponge?
No, I think that might be a tunicate.
It could be. And then
Look at the brittle star.
Ophiuroid above it. And we're good whenever you guys are ready. Navis of Science. What time's our off bottom time? There's a cup corals. Looks like two growing off the same stem. That's
Yeah.
weird.
It wants to be a colony.
Yeah. Not quite. Looks like this one looked dead right here on the side.
Yeah, it's a really nice view of what it looks like with the skeleton and what it looks like with the flesh and what it looks like without the flesh.
Yep.
There we have a nice fan there on the wall.
Go ahead, Nav. 50 minutes. At 3 o'clock. Thank you. There is a white fan. Yeah, right at noon.
Looks like there's
Maybe a. Oh, Stylaster.
Stylasterid, yeah.
Mm-hmm.
Now we've talked about them a few times, but we haven't actually gotten a really good look at one that I
It
can.
looks almost like Madrepora. Very zigzaggy. I don't see
Yeah.
polyps though, which makes me think it's a Stylasterid.
Looks like a Stylasterid for sure.
Then a crinoid. They're light cream color crinoid, maybe a bryozoan underneath it.
Oh yeah. Now Stylasterids are hydrozoans,
Hydrozoids,
so
yeah.
they're actually
Yeah.
related to the jellyfishes. Can we get a little look at that blue sponge up close too? That'd be great.
Look how cool. That color just is crazy. It's very rarely do we get to see blue things.
Yeah.
Go ahead, Nav. Oh, hour and 15. Okay.
There we go.
Thank you.
Nice. It's the best part of daylight savings. You know, we get the extra hour. Man.
Look how cool the blue is.
Now, I wonder if that is identifiable.
It has a, like, its oscules have a, what are they called? Like a ring of, what am I thinking of here?
Well, like a ring of tissue?
Yeah, like a ring of thin tissue on the outside. I know a lot of the times when I'm, like, we're working on sponge identification books with Chris, that ring around the oscule is an identifying feature.
The lip.
So, yeah. So whether or not there is one, a membrane, like a thin membrane, or whether or not it's there or not, or whatever, that can be an identifier from a from a video versus having to actually put the spicules underneath the scope. Yeah, another fairy or something. And a shrimp. And a squid butt.
I think there was a bryozoan tucked in there too.
Another stylaster, maybe?
Yeah. Should we take another zoom in there?
Yeah. There we go, Chrysogorgid. Yeah, there it is.
Nice.
You can see some of the
the little pteropod shells?
Yeah, pteropods, thank you.
That have fallen down from above?
I think it's time for me to have another shot of espresso. My words are not coming to me right now. Yeah, Chrysogorgia, that's cool.
I wonder if we can... So we're going to spend a little time at this wall, think about something that would be valuable to collect. Could we look at that white sponge that's just, what do you call it, 7 o'clock?
There we go, center screen. Very thin. Oh.
Ooh, that's a bryozoan.
I think so.
Wow.
Cool. We've got another coral up here, right above it, and then maybe a barnacle.
Barnacle for sure, in the wall there.
And then again, oh, this looks like Madrepora. If you zoom up, if you go above the bryozoan right there, that looks like Madrepora to me. You can actually see the little corallites on each end of the zigzag. So that's a new species we haven't seen yet, this dive. Very tiny.
Madrepora is a real scleractinian then.
True scleractinian versus the hydrozoan, the hydroid coral, the Stylaster eridae, which is, I don't know, there's different genera inside Stylaster eridae. They're not all Stylaster spa.
It's extremely diverse. It's actually, there are a lot in there.
We use Stylaster like we call Kleenex, tissues Kleenex, It's a very generic term. You can see this looks freshly scraped. This sediment rain,
Sediment's just rained down.
But some more exposed wall up top.
Oh, so people have said they haven't, so far as they know, we haven't collected any of these blue sponges ever.
Okay.
So
I wonder if
we stumble upon one that's kind of
squishy enough to grab.
Yeah, I wonder if with the suction sampler it could kind of
peel it off.
Peeled off a little bit. Yeah, that's how I would do it if I were out there. Suction it with my mouth.
Yeah, if you were in charge of the arms.
I meant if I was in the water. Pull out my dive knife and
peel it off.
Peel it off.
Yeah, that's the species. That one's tiny, though. I don't think you might struggle with that one.
But that is definitely what we're looking for.
With the large oscules and the membrane around the...
Now you can see a similar species that's not blue right adjacent to it.
Yeah, trypophobia. That's the fear of things with holes in it.
Really?
And that's what it looks like. That's triggering my trypophobia.
That's a new one for me. I mean, I respect phobias, but
Yeah. Yeah, go ahead.
yeah, let's take a look.
Oh, those cup coral? Or the blue sponge? Yeah. And then we have another, a bamboo coral, it looks like, at 3 o'clock, coming off the side.
And it is just the color of the sand, isn't it? The sediment.
I think that's our first official bamboo coral this
I think
cruise.
the last time we zoomed in, not too far from here, we also, someone commented that there was a bamboo. So,
Oh, we did see a bamboo earlier? Was that what you said?
someone commented in the,
Okay.
there was a bamboo in the background, just while we were looking over at the other larger blue sponge.
Hi.
Hello?
Thanks for calling the Okeanos. This is Stephanie speaking. How can I help you? That didn't work either.
Worked for me.
Did you tell I worked in retail for a very long time? That's neat. This is like worm, maybe? And some bunch of sponges. There's lots of worm tubes.
There's a coral right in front of us, too, right?
Yeah, what is that? Another bamboo? He just disappears, don't they? They're just like out here. They're the same color as the sand. There you go. Is that a bamboo?
This is coralites.
Oh, it is. Look at the
pulled
base.
in. Yeah, nice. So that's probably the bamboo we've been looking at.
It's yellow.
It is, yeah. It's a little bit of a yellow color. Has some pink polyps.
Is that something that's...
Is that fancy schmancy?
It's probably S1 clade. It's probably related to cladorisis. It's the only one I know that has yellow tissue in this area.
Did you say
All the
S1?
polyps are retracted. Having a little bit of a hard time seeing anything more about it.
Mm-hmm.
That's all right. Cladorisis-like.
Yep.
Wait, did we see a brachiopod in there?
I don't
Robert
know.
Carney was suggesting. I'm trying to...
Yeah, below.
A brachiopod would look like a bivalve mollusk, but instead have... Oh, maybe that...
Oh, this right here.
Yeah.
So right at 9, it looks kind of like a donut with dust on it, like a sugar donut. Yeah. Is that a brachiopod? Looks like a brachiopod.
I would guess so. I mean, can't look inside it to know. But
Poke it.
I think that is a brack.
Mm-hmm.
These are organisms that are, very common in the in the fossil record.
Mm-hmm.
Filter feeders and have become much less diverse over time. Almost at the same time that bivalves have become more diverse. Bivalve mollusks, that is.
55 million years ago, brachiopods appeared.
Oh no, 550.
Sorry, 550. What did I say?
55.
Oh, 550 million years ago.
I thought. Thanks, video. I think we've had it. Yeah. What is that? Take a peek in there.
Sea pen. Yeah,
Is that a pen?
I think so.
Oh yeah, it's sticking out and then bending up. Nice. I think that's a different sea pen than the one. Oh no, it looks pretty similar to what we saw. Or does it?
I think it's different.
Yeah, I think.
The other one, I couldn't see the peduncle at all, but it might have just been buried.
Yeah, I still am not sure. The white one that we saw earlier, I still think that was a bamboo coral. This one,
Definitely a pen.
Subtilum sea pen anchored in the sediments.
Nice looking.
Just before we go, if we can get a look at the very top of it, please. Looking to see if the colony comes to a little point, if the internal skeleton comes to a point at the top. And
I don't.
No, looks kind of.
Don't see that.
Like it's got terminal polyps.
Yeah.
Maybe there's a little in the back there.
Oh yeah.
Okay.
Maybe right.
Yeah. Yeah, I think that's the internal axial rod with a bit of tissue over it.
Uh-huh.
So I did see something like that on the white one earlier, so maybe now you are both convincing me that that white one was also a sea pen, but we couldn't see the peduncle on the earlier one. Great image, thanks.
Thanks, guys. What's happening to that squid? There's another squat lobster. Oh, here we go. Here's a little bit of wall.
Ooh, that looks great.
I see a gorg of some type at 3 o'clock. Hey, did somebody call in?
That's me, Mary Wixton.
Hey, Mary, how are you?
Oh, I have classes.
Oh, yeah? Are you able to watch a little bit?
Yes, you just had a shrimp, a thread-legged shrimp. Nematocarcinus is the genus.
Thank you. Pneumatocarsis. Is that is that one that swims in the water column?
It can.
We have a siphonophore,
Yep.
a benthic siphonophore.
Thanks, Mary.
So it's got...
Yeah, that's going on here. That's crazy. He's really busy.
Yeah, he must have been dislodged. So these normally will sit attached to the bottom with some of their tentacles holding them down, and then not using their swimming bells very much, but a float to keep them up off the bottom. But this one looks like it's sort of unhappy with where it is and is trying to
escape
or just, resituate itself.
That's like a dandelion, right? That's what we call the dandelions?
Yeah, dandelion siphonophore. So,
Ooh. He looks like he's there, though, huh? He's attached.
He's still holding on. Oh, yeah, closer look would be fantastic at this guy. So this is something, from the siphonophore group that we see in the water column quite often. The one that's familiar to most of us is the Portuguese man-o-war. That's at the surface, of course. And these are colonial animals, and there's one group of them that lives attached, very close to the seafloor, where they basically hold on to the seafloor. And
Look at him going crazy.
You can see all of the swimming bells pulsing there, and it has...
Looks like he's having a panic attack.
It
Poor
does.
thing.
look like. It's
So what's this globe on top? Do you know what that is?
Yeah,
The
it's
bell?
called a pneumatophore, and it's a gas-filled float. That's what
Okay.
we call it. And they can control the gas in there, and it helps them control their buoyancy. And so that's what it uses to orient itself upright in the water. Really amazing.
Yeah.
Amazing. Now this is something we could try to collect, but it might come apart a lot, you know, and
You can see the tentacles. They look a lot like the Portuguese man-o-war tentacles. They're like kind of rippled and make little spirals
yes.
off not the ones at the base here, but underneath that.
Mm-hmm. They have tentacles and then side branches, tentilla. And that's what they use to catch their prey.
I want to pull them off the bottom and let them.
Yeah, don't you want to set it free and
let go, bud?
Watch it settle? But it's tied itself into a knot.
Jeez.
It's all tied
No
up.
wonder he's freaking out.
Yeah.
I've never seen one that active before.
No, they're
Usually
usually
pretty chill.
calm, and we catch them while they're sitting in a happy spot feeding.
There's something 9 o'clock of laser. We could get a zoom on that real quick. Right there. White thing.
Got another bryozoan.
Yep, another bryozoan. Okay, that makes me happy. Thank you.
Lace coral.
Whoop.
Wow.
Nice. Lots of squid.
Is that a black coral?
Yep, another black coral. But ugh. I think I need some espresso.
Is that Bathypathes?
Yes, thank you.
See that small animal floating from right to left? Possible to get a zoom on that as it goes by? There it goes. Oh yeah, nice little Sidipidtenophore. I think we can keep moving, thank you. Wow, look at that. That's an interesting Galatheid. Huh. Oh, now I want to collect them both. I think there's one of the shrimp on the bottom. That's, I believe, what Mary Wixton said was Nematocaris. Yeah, we have a chance to check. Yeah, that darker layer looks like maybe it's a bit harder and has probably been around longer for more things to colonize it. So we're looking up where we're headed. Now we're really on to the Scarp-like feature that we expected. Small little larvacean house floating by as we sort of not very consolidated cliff material. Yeah, there's sargassum there on the bottom below that coral. So this is steeper than it was before. Getting a look down the edge where you can see all the sand that's been sliding down from the above. See if we can spot any of that blue sponge to collect. Yeah. Looks like you can see several crinoids on the wall, just passing out of view. Sure. A crinoid and the gorgonian, maybe. Yay! I'm glad the squid are still with us. The ilex squid have been with us this entire dive. That is a very nice red sidipid ctenophore. Don't see its tentacles. They must all be pulled in. Oh yeah, there they are, just at the bottom there, exiting far away from the mouth as possible, practically. There's a large number of these red sidipid ctenophores that are still yet to be described. Wow. Yeah. Take a close look at that.
Looks like
It's
a rock.
Yeah, a little stylasterid below it at 7. That is a nice wall.
That is a sharp wall, isn't it?
Yep.
Really high.
I think we're trying to get closer to it right now.
Almost 90 degrees. Layered.
Really, really cool. Oh, I'm curious.
That is.
Can't wait to get close.
How far away are we? We look pretty close.
No, we're close, but, you know.
No, closer. Look at all the crinoids.
So many crinoids.
Bamboo coral, maybe?
Yeah, should we take a look at that bamboo? Yep, there goes the red sidipid again. Yeah, that'd be great. I think that...
Nice. Yeah, a little, very pink. What is this?
Ooh. I don't know, maybe...
A worm or something there?
A worm, or I was thinking cucumber, possibly. It would be
maybe.
great to take a look at it after we've gotten a good look at this. Cool. It's a bamboo.
Yep.
Mm.
Yeah.
Just above.
And so right there. Okay, so 10 o'clock. It looks like
a worm.
A worm. It looks like it could be a little hairy, like a caterpillar, right above laser now. So it's on the shelf. Yeah. Yeah.
A little bit higher.
There you go. There you go. There you go.
Exactly.
That little bug.
Oh, I think that is another weird polychaete. Oh my gosh. Or
Cucumber? It looks like it has a lot of... No, it's got to be polychaete because it looks like it's collecting sediment, right? And using that almost like a tube.
I really don't know.
I don't know either.
Oh.
Anybody have any ideas? Landside?
Scott says don't have the telecon going.
Oh. The teleconference call's offline?
Yeah.
Okay, hang on. Well, I'll get that fixed.
There's the blue sponge straight ahead, about 7 o'clock. Not sure if that's much bigger than those, but also small. All right, let's not. Yeah, yep. "30 minutes left on the bottom." I think we should get another water sample and then one right at the end since we have two. I know, it's really easy to imagine. 4, I think. Mm-hmm. Great. Oh, that does have a nice blue sponge, yes. Oh, it's sticking up. I think that's a good choice.
The conference call has been reconnected.
Conference.
Oh, welcome back.
Sorry to not know what you were asking for, Scott.
No problem. I was persistent.
So Chris Ma was able to identify that interesting sea cucumber. Yeah. Olafinius. With a GH. Olaf. GH in English. It's always a... All right. Now, while it's out there, she just hoover. When will we be back at this wall? Actually, this wall doesn't seem like it'll exist, right?
Alan, so you're about to collect the blue sponge, is that correct?
Yes. Is there anything else nearby there that... While we're at it?
I don't know if you earlier in the dive saw any of the yellow fans, but I noted there was a yellow fan above it. So just to get a look at it to see if we can get it to genus or something. Thanks.
Absolutely. See if that's Paramuricea or...
I think we only have about a half hour left. Just a heads up.
Yeah, Paramuricea canthogorgia.
Mm-hmm.
Yeah, it's right above the sponge. You don't have to move or anything.
Yay. I think all we're going to get is Piscilla sclerata.
For the blue one?
Yeah. So the substrate is likely to break pretty easily.
You should be able to like kind of peel him off the rock, and he's probably going to be very soft.
Yay! I'll take care of the worm.
Please take care of the worm. I can't even stand looking at it in the camera. It like freaks me out.
It's going to be so great.
Ugh. Be careful though. I don't want you to get stuck because sometimes they're really venomous.
Uh-huh.
Poisonous.
Poisonous. There
Ooh,
it is.
there he
Yay! Success.
So we think that this has never been collected before. Is that the thought process?
Yeah.
Awesome. Did he get in?
Which did he go into?
2. Bucket 2.
No, he went... Oh, shoot.
Oh, and just... Oh, okay.
Yeah, I put the wrong thing.
That was bucket 2?
Yeah.
No, but can we get a good look at that yellow coral that's right in front of us? Yeah, no worries. That's good.
Oh, it is so great. Sorry, that was me looking at the scale worm
Ooh.
inside the canister.
There's lots of ilex squid, huh? We think we're going to put that annelid in formalin?
Yes.
Just let the sample data managers know.
Mm-hmm.
So the last sampling we did of a annelid, or the way I was taught in grad school, which was not that soon ago, was to cut the head off, put the head in formalin, and then put the rear end in ethanol for DNA. So we do use the rear end for DNA in the face because a lot of the identifying features are in those things on the head side. Whatever they are. So tentacly things on the front.
Yes, exactly.
That's cool.
Ooh, this is a nice
Gorgonian.
Yeah, from this distance, this looks like it's probably in the Paramurcia group.
Paramurcia.
And not Acanthogorgia, but stand by on the zoom here.
Okay. He's very fuzzy looking. Like maybe,
spiky.
Spiky,
Yeah.
spicular spiky.
Yeah, that stickiness, actually, maybe this is a Acanthogorgia. It's got a clear anthostele. That lower part, it almost looks like a cup that the tentacles arise from. Contract into that, but the Acanthogorgiids have a very thin skeleton. Yeah, now I'm thinking this is in the family Acanthogorgiidae.
Awesome, thank you.
Cool.
We're good, guys. Thanks.
Really nice. Thanks again for that collect, if we didn't mention it.
Another little shrimp there. 9 o'clock. Maybe another bryozoan, a little fan. Little mesh fan.
Yeah.
Yep. Maybe not. Oh, yeah. Bryozoan, yeah.
Yeah, that bryozoan's been really likes this place.
Oh, Stichopathes. I think that's the first one we've seen. Yep. Still pretty sparse, though. I think maybe a Chrysogorgid at 6 o'clock. Yep, there you go, right there. Oh, no, maybe not. Yeah. Pink fan. Yeah, bamboo coral. You can see the nodal branching. That's neat. It looks like maybe another bamboo coral right above it, or around 2 o'clock. And then we have that black coral, the whip, coming in at noon. This wall is still really steep here. Nice. So that's a really orange Stichopathes type of black coral.
It's just really bright.
Looks really healthy too. He's totally not sloughing off any tissue.
So the very thin coiling whip in front of it.
Yes.
Does that have octocoral polyps on it, the white one?
I'm not sure. Guys, can we swing? Yep, can we look at that white whip that's to the left? It's very tiny. Yep, right above the left laser. Yes, it does have polyps on the tip. Looks like it could be a bamboo coral. but it looks like the nodes are almost
Definitely.
gold. Right? They're not really that
They're
brown.
really, really small space between those nodes.
Yep. Yeah, definitely bamboo, and I see those golden nodes. That's kind of interesting. Well, that's good. It's also really short internodes by the looks of things.
Mm-hmm.
An odd one. Thank you.
Yep. Then it looks like we have another bamboo coral at 3. This little rock here looks like it's about to fall. Right there.
It does.
Give it a good
don't
kick.
Don't fly under that.
Yep. We've got a good bamboo coral, I think.
Yeah.
I would like to keep moving.
There you go.
Ooh, very silty sand. Thrusters cause that.
It does.
Yeah, it's very fine, silty sediment. Oh my goodness.
It just keeps going up and up and up.
Yeah, we're still at 900. The never-ending, like, Twilight Zone depth. It's neat. There's like a really black layer just right in the middle there. 10 minutes. Okay, thank you. Maybe Chrysogorgid. I think that's what that is. Yeah. Chrysogorgid, another blue sponge. A couple of yellow sponges and white sponges in there. I can see how loose this looks. Like this could just go at any minute, right? That really, the layering. Like
Yeah.
I feel like I could put my hand in there and just pop that whole section of rock right off. Just
Can we
flop
bring
down.
it up and then
Yeah.
sort through the organisms?
Yeah, of course.
So it looks like that ledge where we're seeing things is darker in color than what's around it. So I wonder if it's either a different mineral or it's been manganese encrusted, which is making it a little bit more stable. That's why there's some fauna growing out there. That's without knowing any geology.
What is that?
That certainly makes sense.
What is that? A siphonophore.
Oh, it is a siphonophore
That's
drifting
huge.
towards us. A long one.
Whoop, not anymore.
It said, I don't like what's near me.
Yeah, a cloud of schmutz floating around.
That was really cool.
If you run across any Chrysogorgia that have squat lobsters on them, that would be interesting to me.
Yeah, sure. We just saw a Chrysogorgid, so maybe we can swing back and look at it.
Wow, and its back stretched out.
That's neat.
Great example of how these guys
Whoa.
He's a couple meters long.
And then he's back together. Whoa,
The squid
and a
coming out.
squid. So many.
It's like a pogo stick.
You asked and you received. So it seems like it's done that 3 or 4 times. Oh, there's a nice close-up of its stem and all the different parts, all the different zooids. And those yellow parts, I believe those are the tentili that it's...
Ooh.
Whoa.
It curls.
Yeah.
Very springy.
Yeah, I think that footage is good enough for someone to identify this species.
You can see the bells moving at the
Oh,
top.
there you can see the tentili. They're dangling from the tentacles, the little smaller yellow bits.
Mm-hmm.
So the larger yellow bits are most likely the gastrozoids, the feeding zooids.
Alan, I think I noticed 1 or 2 of those zooids was really large, and I remember a paper from Steve Haddock's lab that showed that the feeding zooids could ingest larvae, I think it was, 3 to 5 times the size of the polyp, and they just, expanded in size to accommodate it.
Cool.
So I wonder if that's what we were looking at there.
Good chance.
Yeah, can we get back on that chrysogorgid?
As the squid patrol goes by again.
Mm-hmm. This cloud of dust. It should clear out soon, but yeah. All right, Mary, we're getting close to zooming in on it. You've got a bit of a delay on your end on the video, so just give it a sec. We should be zooming in a little bit. There's also another squat lobster
On the rock.
at 8 o'clock on the rock.
That's the right squat lobster. Can you nab him?
Okay, you want us to grab one of these squat lobsters? Can we collect this chrysogorgid?
Yes, please.
Not sure we have time, Mary.
Well, if you see one in the future, please grab it.
You're interested in the one specifically on the chrysogorgids?
Yes, please.
All right. We've seen a few of these this cruise, so I'm sure we'll see more. But if we see one again, I will definitely grab it for you. Actually, we collected one, didn't we?
I know that we got a Paramaricia with one. No. That was with the Astroschema. Yeah, I think we did.
We got a chrysogorgid.
Maybe right at the very beginning.
Hey, Mary, I can send you a picture of the one that I think we collected earlier in the cruise if you want. But we can still get another one if I'm wrong. Just that we're really ending up close to the end of this dive, and so. Just have like 3 or 4 minutes left.
Yeah, I'd like to see the picture.
Yeah, I'll get them to you. Do that after we're done sampling today. I'll email it to you.
There's another squat on the rock. Next.
Yeah, it's a
Next
different
to this
squat lobster.
bamboo.
Yeah. He's very armored.
So this,
Go ahead, Scott.
are you able to get a little closer? Yeah, it looks like there's a couple of very weird places. You see those weird twisty branches just went off the top. There's another one there. I'll bet there's an anemone right in the center. We've been seeing this when anemones settle on these corals. They then cause subsequent growth of branches to be very aberrant relative to the rest of the colony.
Odd.
That uppermost one, if we can get a zoom on. If you have time, I realize we're very close to the end and want to move, so don't worry about it.
Yeah, if you can zoom right
That
there.
squat lobster is Immunodopsis.
Immunodopsis.
Not a chirostylid.
Thank you.
Now look at
Hey,
that branch.
fantastic zoom. There's definitely something in the center there. You can see there's a pinky blob.
Yeah. At like 3.
I wonder if it's the backside of an anemone. So the reason I'm really excited to see this observation, we were seeing a lot of that, especially this summer, and we've seen some in the Pacific, and we're starting to accumulate enough observations to be able to determine, is this only happening in one species of bamboo with one species of anemone, or is it a more broad-scale phenomena? Great. Thank you so much.
Yeah.
It's like the presence of the anemone sends the coral haywire, and it starts putting out branches in various directions, and it creates kind of a little cage around the anemone almost.
Huh. Okay, that's cool. You spot an amphipod in there? Yeah. On the branch to the right.
All right, we're good whenever you guys are.
The alternative, if that wasn't an anemone, I suppose it could be like an ascothoracican under the tissue. So kind of essentially like a relative of a barnacle parasite.
It looked pretty soft. I would have called it the foot of an anemone, just that the anemone's tentacles were tucked or not facing me. It looked like that, like little squished, retracted guy.
I agree, Stephanie. I think that's what it is, and that would certainly make sense with our previous observations.
Whoa.
Could this be the fabled top of a wall?
I don't think so.
No.
That I've often heard of but never seen?
We have another like 70 meters to go before we hit the, right? Was it 8:30 or something?
No.
8:20. Yeah, forget it.
Yeah. So as far as we know, based on our data, tops of walls do not exist.
Yes, correct. Or we are just incapable of getting to them. They're like in the 5th dimension or something.
It's a good thing the walls themselves are so interesting.
I know, right?
They are.
Yes, please. The last Niskin bottle would be great. That's going to be sample 7.
Thank you very much.
Okay, thanks.
Thank you so much.
What number was that? That was 7W.
7W,
Okay.
yeah.
So we're ending this dive at roughly 892 meters. We started this dive. Oh, so, okay, first we're 30 miles south of the dry Tortugas. And if you'd been following the cruise but didn't tune in yet today, we had to come off the Blake Plateau the past 2 days and escape some gnarly weather. So we've come down to the south of the Keys. So we're diving right now in Tortugas Valley. In 1,000 meters of water is about where we landed. And the plan was, based on the multi-beam, to go up a really tall wall. We didn't really find that wall until, I don't know. It feels like 20 minutes ago. It's probably about a couple hours in. It took us a while to find the wall, which is...
Oh, many hours in.
Yeah, it was many hours in. So the wall wasn't where we were expecting it to be. So the thought process is that it sloughed off between the time the multi-beam was collected roughly in 2014 until today. So I think the plan is tonight to just run a run
Another
a multi-beam
multi-beam.
over it and see how it's changed. Yeah, so because it looks like it just fell. The whole
Yeah.
thing just like avalanched down. Because we were supposed to get to 820 at the top of the wall, but we were at like, where we ended, 9-something, halfway up the wall. Yeah. And we should have been at the top of the wall like hours ago. So it looks like a big chunk of it fell off.
I know.
Yeah.
Which is not surprising when you see how we could see its, small slides. It's not very consolidated.
Yeah.
So
It's that it looked like that soft clay, or really like not quite solidified hard bottom. It was more of that like clay, malleable almost.
yeah.
We picked something similar up in the sinkhole on the far east of the Blake Plateau. What day was that? Friday.
Mm-hmm.
So when we got down, let's see, it was mostly sand, hardly any waves. It was mostly bioturbated sand. So the so that means there's fauna or stuff growing inside the sediment, or causing divots and mounds to form.
We saw a lot of mobile fauna on top of that sand, so especially this ilex squid,
really
which was
common.
with us from the beginning of the dive all the way to the end,
Yeah, absolutely.
as well as many different kinds of fish,
Yeah, we had a lot of fish today,
eels,
too.
and some really great tripod fish, 2 different species of tripod fish.
And then when we finally reached the wall, we did find some corals and some our typical, hard bottom fauna that we see at this depth.
Encrusting demo sponges.
We collected 5 water samples, one at 500, one at 1,000, one at the end of the dive, and then 2 in the middle. We also collected this really
Beautiful scale worm.
Oh, they freak me out so bad.
Not everyone agrees.
Yeah, I know. And then number 6 was the blue sponge.
Yes,
Sample 6 was
Pisciclera sponge. At least that's the hypothesis.
Yep.